Hi, welcome, and thank you for joining us today from all across the country and beyond for our CFD Training Center's first Decoding the Data Ecosystem virtual symposium. My name is Jennifer Burnett, and I serve as the director of the CFD Training Center. The Training Center is managed by ORU in coordination with Biodata Sage LLC. Today, we will explore emerging tools and resources, hear directly from CFD experts, and discover innovative projects demonstrating how CFD data can drive discoveries across disciplines. The Common Fund Data Ecosystem, or CFD for short, includes a wide range of diverse and valuable data sets for omics-related research, including genomics, transcriptomics, proteomics, metabolomics, and beyond. Whether you're looking to integrate real-world data into your curriculum or discover new research directions, this symposium will offer practical insights and community connections to help you get started. Before we jump into a few housekeeping items, I do want to take a moment to thank the entire Training Center staff and our partners at the other CFD centers and data coordinating centers for your support in this event. And a special thank you to today's presenters from across the CFD ecosystem. Now for a few housekeeping items. Bo, next slide. Well, brace yourself, the conference is here. You can keep going. There we go. So a few housekeeping items. Please note you can access the full schedule on our website or through the QR code you see on the screen. Due to our packed agenda, there is no formal Q&A today after each presentation. Questions are, however, welcome anytime throughout the presentation and will be answered live if we have time, or in writing if not, through the Q&A function on Zoom. Closed captioning is available through the Zoom platform if you need it. Please also note if you have to step away or miss a portion of the symposium, a recording will be available to everyone who registered, and we'll hopefully get that recording out to you within the next week or so post-event. With that, we'll move into our opening remarks from George Papanikolaou from the National Institutes of Health. So George joined NIH Office of Strategic Coordination in 2021 and currently serves as the program leader for the Common Fund Data Ecosystem and the Newborn Screening by Whole Genome Sequencing Collaboratory. He previously held the position of program leader for CFD's metabolomics. George has a PhD in human genetics and acts as the program officer for the CFD's Knowledge Center and Cloud Workspace. Welcome, George.
Thank you, Jennifer. Hi, everybody. I'm having problems with my camera. I will try to fix it later, but thanks all. I'm going to turn it off now so I can actually get this presentation going. So I wanted to thank the organizers for inviting Common Fund to present today. And I'm going to be talking about Common Fund, the origins and goals of CFDE, and how we directly fund external partners. So we could start the slide deck. All right, if we could skip to the next one. All right, this is what I'm going to talk about. Let's go to the next slide and actually start talking about things. So let's talk about the CFD program team. And allow me to introduce the team who, along with the DCCs, or the Data Coordinating Centers, you'll be hearing that a lot today, that acronym, and centers make CFD possible. Besides roles in centers, we often act as POs for the CFD coordinating centers, the RO3s, and the partnerships, which I'll briefly mention. I'll start with Christy, who serves in the pivotal and essential role as our program manager, as well as PO, or program officer, of the Data Resource Center and the Integration and Coordination Center. Chris, below, is our CDR Assistant Director, who provides guidance to CFD and other programs. And on this slide, we have Renee, who is our program specialist, who helps us with requests and coordinating meetings. Next slide, please. Sahana, who I have the privilege of sharing part of the program with today, has been involved in a myriad of CFD's training efforts and is the PO for the Training Center, which is putting on this nice virtual event. She'll be talking about engaging with NIH later in the day. Haluk was part of the original group that managed CFDE and continues to provide input on CFDE and leads the Common Fund's Bridge to AI program. And finally, Laurel has been involved as PO for our short courses and has done extensive work in Bridge to AI. Next slide. So, brief introduction to Common Fund. Many ideas can be envisioned that are trans-NIH. Common Fund programs have additional criteria to define which programs are suitable for Common Fund support. So they should be transformative, meaning they have an exceptionally high and broadly applicable impact. They need to be catalytic. Therefore, they need to have a goal and clear deliverables that can be achieved in 5 or 10 years. And again, a reminder, common fund programs have a defined period of time. They should be synergistic. They should add value to multiple ICs. They should be cross-cutting. There needs to be a reason why strategic coordination across multiple ICs is necessary. And finally, they really need to be novel. Programs should provide new solutions to specific challenges. Next slide, please. Here you can see many of our programs arranged by the goals that they have. Common fund programs have many goals and activities that span multiple categories. And Noel will later be talking about some of the programs that we have in Common Fund. I'll be addressing the perspective from the Catalytic Data Resources Group, which I'm in, and the rest of the CFD team is as well. Many Common Fund programs create data resources. Even though some of these programs are in our other sister branches, they have secondary assignment to CDR because we oversee these categories. We manage these programs collaboratively, and we want to make sure that the Common Fund supported data resources are well managed and have the broadest possible scientific impact in the biomedical and behavioral research community. Next slide, please. So here we view sources and types of Common Fund data. Our programs produce rich multimodal data and at the individual organ and cell level. And they also go across species. And these programs include foundational molecular studies and clinical research. And the word cloud in the center represents the kinds of data across our studies. So there's plenty of data that you probably would love to get your hands on. And today we're going to talk about all the various ways we can help you do that. Next slide. So looking at these Common Fund transformative foundational data sets, the question we have is, when we're envisioning the Common Fund data ecosystem, is what transformative impact could be achieved by uniting these diverse resources? And that question inspired the creation of an ecosystem, which is CFDE, what we're here to talk to you about today. So next slide. So let's focus on what CFD does. The Common Fund data ecosystem provides infrastructure needed to create a functioning ecosystem that makes Common Fund data sets more impactful by enabling easier data access and use. And we have 3 key goals. We want to make sure that you can search across and use Common Fund data sets. We want to sustain Common Fund data sets and products after those programs end. And we also want to train users to work with Common Fund data. Now in its second phase, CFD continues to adapt to support our mission, including a new multi-center approach, replacing our previous single coordinating center. Next slide. So whenever we're running programs at NIH, we really want to address those questions. How do we know we're doing the right thing? So what does success for CFD look like? And our chief metric of success is at CFDE is discovery. If CFDE is successful during the 5 years of phase 2, more investigators will be using Common Fund data and products for novel discoveries and innovative purposes. So everybody who's here on this call, your achievements are the cornerstone of what is going to measure our success and how we're going to be successful. And we've pursued that by having 5 different domains and having a center coordinated, a center attached to each of these. So they include data, knowledge, training, workspace, and coordination. Next slide. So one of the reasons we have to assemble this the way we did is we are an ecosystem of federated data. So we have up to a 10-year funding limit, and we have no dedicated Common Fund data repository. So therefore, we are supporting persistent identifiers and data repository services at multiple DCCs to provide a standard way to retrieve a data set and elements regardless of the repository's underlying architecture, either at the DCC servers, which we'll be talking about how, for example, the DRC pulls in that data, or the final deposition repository. We're going to hear about sustainability later, and that's one of those things when we branch into that new phase of a program. However, as data enters a general repository, annotation richness is lost. So we hope to minimize that loss through an integration of search, metadata, assertions, and knowledge. And you'll see that in our discussions of what the various CFD centers do and how we're approaching those goals and sustainability. And we have 4 models of common fund data sustainability. Again, we're not creating a data repository. So an IC can step up to maintain a data set like GTEx on Anvil at Genome. And I'm sure many of you have heard of GTEx, and we'll be hearing about that later in Noel's talk. Data generators can receive a competitive award to continue the resource, which was done, for example, by the Metabolomics Workbench. Data can be transferred to a public repository, such as dbGaP, which I'm sure many of you are aware of. And if need be, we could store highly used data. For those of you who've been involved in cancer genomics and molecular medicine, we're talking TCGA level 3 or 4 data in the cloud workspace. So this would be, for example, a list of specific variants, but not having the genome if one was doing genomic research. Next slide. So we have a very pragmatic approach to standards. Next slide. As the well-known XKCD comic points out, the drive to create a universal standard for any data type leads to yet another standard. Next slide. In CFD, we have taken a pragmatic approach by favoring existing standards. Could you forward again? The DRC uses FairShake for fairer evaluations, Croissant to produce common fund data for machine learning use, and a CFD partnership has updated GTEx's widely used RNA-seq pipeline to harmonize our existing transcriptome data. And we have that pipeline available for anyone who wants it. Just contact me and I'll put you in contact with the people who have it. Next slide. The Ontology Working Group, chaired by the DRC, which we'll be talking about a little later, has members from each DCC and has identified control vocabularies that cover the wide range of common fund data, from molecules to cells to individuals and across species. And these standards from the Ontology Working Group, as well as our metadata standards, C2M2, these are things that we would love to share with you. We've got them to work, and if you look at the various consensus metadata standards, if you can get someone who's interested in mouse genetic expression and somebody who's interested in protein and metabolomics and get them all to agree to controlled vocabularies, we think we have a pretty rigorous approach. And again, we're more than willing to share that. Next slide. So where is CFD today? We have 5 CFD centers and 14 common fund programs that are building the data ecosystem. We've had 16 partnerships to date, and some of that information will be shown later in our talk. We also have partnerships are available to external members. I'll talk about that real quick. And we've had 62 pilot projects that enhance the utility of common fund data. Next. So we have additional support for common fund data users, as I mentioned. Next slide. And we've had partnerships. We funded them for 3 years. We have a goal of returning to this project for another 2 years. And these partnerships have at least 2 or 3 common fund DCCs creating tools along with outside team members, then those are required so that we can bring in some innovation. Things that we've produced include the RNA-seq pipeline, the data distillery knowledge graphs, and the playbook, which we'll be talking about later, which allows you to do computational visualization of data through APIs. Another thing that we've provided every year and hope to again is RO3s that require incorporating at least 2 common fund data sets. They are 1-year 200K awards. They've been in the past, and up to 20% of that can be used for wet lab work to confirm results. A great way for someone who wants to prepare for an RO1. And because these awards are small awards, they don't impact on somebody's ESI status. So that for people who are starting out as investigators, ESI status is something you want to maintain so that you can, is more helpful for you getting your first RO1. The products are returned to DCCs, repositories, and GitHub. And again, we've funded 62 of these to date. So it's a great way to tip your toe into common fund data and bring your own data along with it. Next slide. And We have some new partners that have come in. You can see some of this stuff at the DRC's info page, but what they're doing is really building tools or resources for the community. They have specific use cases and demonstration projects to do that. Glad to have you look at the DRC and explore more of those. Next slide. And just again, mentioning the team from CFDE, that QR code on the right is where we announce our funding opportunities. And I will work with the training center if we have new funding opportunities, and we hope to soon to share those with you so that you can also engage with CFDE and have us fund some of your research. So thanks again from everybody from the CFD team and for this opportunity to talk about the program.
Thank you, George. All right, next we'll move into our first session of the symposium. We invite Noelle Burt to get us started with this Navigating the Common Fund Data Ecosystem presentation. She is from the CFD Knowledge Center, as well as the Director of Operations and Development of the Diabetes Research and Knowledge Portals in the Medical and Population Genetics Program and Metabolism Program at the Broad Institute. Her work centers on operational and organizational leadership of large-scale international genetics consortia and public-private partnerships for human genetics studies. So welcome, Noelle, and thanks for joining us.
Thank you. All right, do you mind if I share my screen?
Go for it.
All righty, beautiful. All right, lovely. So I want to thank the Training Center for inviting me. I'm so happy to be here. I love talking about the CFD. And George, that setup was excellent. And I might overlap a little bit, but hopefully you all don't mind because it will be in the context of a member of CFD, a member of the Knowledge Center, and also an investigator herself, a geneticist, as Jen pointed out. So the reason I want to, the framing I want to give you today is the beauty of CFD, as George illuminated, is I think it's a very unique model. It's a unique model both inside of CFD in terms of how we all work together, but also a unique model for how we might collaborate across all the other NIH programs. Because as you can imagine right now, there is a universe of web-based resources for omics and beyond that we all want to be taking advantage of. And so I think the talk today is really meant to think about how we would all do that, because together with the CFD is sort of at the center of that. And the reason I want to do that is because the Training Center did this really nice survey along with the registration as to why are you here? And I thought it was really illuminating because, 50% of you at least wanted to learn about the CFD tools and resources. So that's great. You're going to learn about that momentarily. But also other topics are really important to you, like collaboration and new opportunities for partnerships and funding opportunities. But then some really cool specifics came out as you were talking about what you really wanted to learn and take out of today. And some of the themes and threads I want to pull together here to set us up for the rest of the conversation is that I heard things like AI tools for health. That's what people want to build. Also, one investigator said, my research relies heavily on web-based omics tools to explore. And I think that means the CFD is a great place for you because we pull it all together, all these 18 DCCs and 5 centers, we're pulling them all together into enabling one place and one sort of interconnected access point through what you're going to hear about today. And I love this last one. This person wants to contribute to the development of fair, aligned data standards and tools that support interdisciplinary research. And that's really what we're trying to do, both in the CFD as a community and beyond. So with that, hopefully this is an invitation to you to come explore the CFD with us, and hopefully the rest of this conversation, which I'll be doing with George, will help you orient to the CFD. So I'm assuming you know about the Common Fund. You also know a little bit more because of George. I'm sure you can probably name a few programs, but really, I don't know if you know how to navigate across them easily. I think that's potentially tricky because there's a lot, right? But also how we are as centers and DCCs trying to create connections amongst our different data types and data resources and analysis and tools to create more meaningful and new connections to enable discovery. Because as George said, that's really what matters at the end of the day. So today I hope to give you and hope you walk away with a sense of what the Common Fund data ecosystem is, which you learned from George, what's unique about our model and how we're structured, which we've talked about already, but 2 important things, how through integration and access, we hope to drive discovery, and how this very unique resource may be useful to you because you may yourself work with your own resource that you think is really important and you might want to connect with us, or you want to figure out how to better make use of all these tools in a more efficient manner. So as George mentioned, the Common Fund is catalytic, and I think this is just a couple of themes to mention. The major important thing is high risk, high reward. They're meant to lower barriers, right? That's what they are as the Common Fund. And you saw this from George, there's a lot of resources. And I imagine as you were listening, you were like, wow, there's so many, they kind of appear unrelated. And beyond these resources, each of the DCCs have entire communities around them that are generating data, contributing to these data coordinating centers. And therefore, they have their own unique cultures and communities. So that's pretty massive, right? But to start, I want to walk you through a couple that you might know, a couple you might not, to give you a sense of the ecosystem and then talk about how it is brought all together. So a staple in the genetics community, you will probably all have used this. This is GTEx with over 3,000 publications and counting. It's a staple, right? It's the place you go to look up your favorite genes, gene expression, and healthy tissue, right? So this is a Common Fund program. You all know this one. But here's one you might not know of if you're a geneticist, for example, which I am, right? It's called Spark. This is really a place where folks who study electrophysiology and how the brain connects to different organs and the electrical signals that travel through that space and what they do is what this resource does. It creates maps for you and spatial assessment of those connections between the brain and the organs in the body in terms of what they function to help alleviate neuromuscular disorders and other things like pain. So that's an entire resource called Spark. Another one that you may be familiar with is HubMap. This to me is the go-to most beautiful visualizations I've seen in a long time. Single cell data for healthy tissue, right? And so this is a resource that allows you to download, visualize, navigate through single cell data. But more importantly, it also has an entire ecosystem itself of investigators around it contributing data and also conducting research on the data within HubMap. And so that level of knowledge and results also is something we want to bring together and leverage within the CFD. Glygen. If you're a biochemist, you know this resource because you go here to understand if your favorite protein is glycosylated. But did you know that there's a whole host of computational approaches in informatics and aggregation that this resource does that can be not only useful to you if you're a biochemist, but if you're a human geneticist or if you study a biological process. XRNA, another one. I could go on. But what's great about this is another resource that allows you to see, extracellular RNAs aggregated across data types. But also the consortium surrounding this has enabled things like Discover for Preeclampsia, a preclinical biomarker that helps treat and triage patients with that at earlier stages. So again, access to human health through these data coordinating centers. So George showed this. And I can imagine you saying, what is the benefit of bringing these resources together? Well, I can imagine I'm hopefully convincing you of that. But there are some themes and threats that bring them together, right? They're all data aggregators. Here's an example. Glygen is one. GTEx is another one. They're all creating reference maps, right? You have HubMap that I mentioned, right? Molecular signatures are another theme and thread. You've got links is another one that I mentioned. Motor pack. I'll talk more about these. These are creating these molecular signatures and making them accessible. And these themes and threads are not only to one DCC, but across many. And they all have these core values. All fair. They all have their own communities around them, and they all really partake in engagement with the broader community, learning and adapting and making things accessible to others. So with those core values and those themes and threads, it really is quite easy to see how you can actually create an ecosystem around them. So George showed this slide, but I think it's really powerful to show it again because what we aim to do is create an infrastructure to query across these resources and make them more useful to each other within, but also beyond. Sustain and maintain this, right? Because these will not live forever, as George indicated, but if we can create an ecosystem and create representations, summary representation, APIs, different ways of accessing them, they also live forever because they're used over and over in different ways. And ideally, in order to make this useful, you got to train everybody to use the data. So with that, I thought I'd flip to how then do they all connect? So here's an example. So there's data within Glygen, right? That is useful only to glycoscience, but everything, for example, connects to genes, and genes are informative to inform on biological processes, right? So Glygen may not immediately click to you like, oh, it has information on genes, but it does, right? Because it's connected to a protein. But within the ecosystem, there are 14 other resources that are connected to genes. So you can imagine that anchoring to a gene is a way to navigate across many of these resources. Another example is GTEx again, right? This is the go-to site for gene expression. But did you know that there are 10 other resources within the Common Fund data ecosystem that have gene expression data? So not only if we brought all this together, as George mentioned in his pipeline, all these together, you could run differential expression on all of them, and you could learn not only in the healthy GTEx tissue, but in some of these disease-specific tissues that these other Common Fund programs have generated. So putting that all together, one pipeline, making that accessible to you is really the benefit of the resource. And I'm going to show you a little more about that. So pulling it all together with 5 centers, as George mentioned, I'm not going to spend too much time on this because we're going to talk about them a little more. And it all ties together into this lovely ecosystem. Interdependencies, but all having their own programs and functioning on their own. It's a really unique model. I think it's really fantastic because it creates a sense of community within and broader community, which is really what we all want to be part of as a community of scientists. So this is what it all looks like. Again, these 18 coordinating centers, all representing different omics, and these centers at the core of it, really trying to create connections and support the work of these programs. So I'm going to talk a little bit about the two of the programs today, but luckily I have George to help me with that. So we're going to talk a little bit about the Data Resource Center and the Knowledge Center, because these are 2 major ways to enter the ecosystem, because each of these resources have taken a different approach, right? The Data Resource Center really focuses on the data and the tools in the CFD. The Knowledge Center focuses on the results and the knowledge generated. These obviously are interdependent and interweaving, but to take those perspectives is really useful because I think scientists, right, kind of think a little bit. Some scientists are like, I want the data. Sometimes scientists are like, I want to see the results first and do some exploring. But what's the beauty of this? These over time are going to become closer and closer together and really navigate into one really sort of front end for you as a user to navigate through. Or of course, you can go to your DCC of choice. So with that, I'm going to hand it over to George to talk about the DRC, and then I'll be back again to talk about the Knowledge Center.
All right, thanks. So the DRC is responsible for creating and maintaining the CFD portal that enables users to query and use data sets from across Common Fund programs. The CFD portal, created by the DRC, will include a landing page for the overall CFD program that integrates information and products across the other CFD centers. So this next year, where it's a real big building year of how to bring all these centers together through a common interface. And here you can see what the DRC really does. They're working with the Common Fund Program DCCs to bring in data and metadata and code. And we'll be talking about the uploads and what the matrix looks like, which is a really great way of exploring all the resources we have available for you. That data, metadata, and the code matrix is wonderful because other people, including the DRC, and people have been able to use that and integrate that to create a series of things, such as the playbook workflow builder, which I'll talk about. That's a great thing to add to whatever site or things you're doing. And I'll also mention we've integrated things like the knowledge graph from the data distillery, which worked across all of our DCCs to integrate that knowledge. The PIs are Avi Mayan, who is at Mount Sinai, and Shekhar Sobranian, who's in San Diego. Next slide. So here we see what the workbench actually looks like, and it's a really great way to get across and as a first way of getting into a website. And again, this is the information portal is at, we'll put it in the chat, is info.cfd.cloud. And this overall picture gives you a lot of information on the CFD workbench. If you saw that wheel earlier, that's because this is something we're starting to implement. If you look over to the right, you'll see the navigational wheel, which is going to help us go from side to side, from site to site, and really allow us to interact at all different levels. We'll also be talking about the data portal, and then, of course, you can see the knowledge portal, and that is where Noel is going to be addressing that. So next slide, please. So one feature that really sets CFD apart and has been part of it since the initiation is the cross-cut metadata model, which is a really great way of structuring data across these diverse studies to allow your data to be integrated and also be searchable and findable. So this is something you can see the reference in GigaScience below by Dr. Charbonneau, but it's really a great way if anybody else would like to integrate their data into this same kind of structure. And as I mentioned earlier, we've done a lot of things with ontologies and bringing them in so they're widely adaptable. So this is a resource that we've been curating and creating. And we can give you more information on that. But if anyone's looking to integrate a lot of different kinds of information, please reach out to us. And and this is our underlying structure for the metadata. Next slide. So this is an example of the data portal where you can actually do things. You can do query. I was helping a colleague this morning, said, I want to know where all the lung data is in Common Fund. We went right here and he said, wow, that's a lot. And yes, it's true. And we'll be talking about some of that, but that's another part of the workbench at the DRC is really a description and finding all that data and information on it. Next slide. So one of the things that we've been really happy with the DRC is its ability to allow for very easy submission of data sets. And their submission portal, and they also have documentation about the API, is really how we get that data in. And it is both rigorous and flexible enough that you could actually have people who have very small defined data sets. And then you have people, for example, like Kids First, which are always adding new clinical studies, as well as subsets of information on patients. And again, every quarter, they're able to adapt and bring that data in. So this is a very flexible system. And a great thing that we do is we upload the data, they're evaluated for fairness. Noel mentioned that's one of the things we really want to do. And I know that some of you are very interested, as you mentioned during the survey, about making your data fair. And we use a tool called FairShake. That's also documented there, and our approach is documented. But you can see our improvements over time, making our data more and more fair. And the individual studies use that information to make sure that their data becomes more and more fair as they go on in their program. Next slide. So here we can see the data matrix on the left, and this is a really great resource to see everything you want to know about a study. So it includes the data sets and metadata, but it also includes code. So if you want to find API to actually interact with some of the resources provided at the various Common Fund programs, you can actually have API code there. We're also providing data on models. That's one of the big things we're going to be doing in the next year, is making sure that more and more of our data is available for AI and machine learning. We also have data in various formats. The final thing I want to mention, I know a lot of people are using knowledge graphs or even wanting to use RAG data to actually work on their knowledge graphs and to integrate them into generative AI, and that's available as well. To your right, this is an example of what happens when you search for liver. So not only do you get the C2M2 metadata, but you can see the data that's actually been processed, and it goes across and tells you, which studies have it, your diseases, your phenotype. So it really gives you that degree of information and that ability to go across. And that's one of the things that the C2M2 metadata model that we're using allows to happen. Next slide. So here's an example where you can actually look for the differences of the C2M2 data for liver within Workbench. Again, a great resource. You're not going to find a better compendium of metabolome data in the world than that program. But if you go to the right, you can actually see the process data, whether it's something from anatomy information in a knowledge graph to actually having something that's been run across a gene transcription tool. So it really points out those things that are not only created by the study, but also things that have been derived and are available for other people. Next slide. So as I mentioned, one of the goals of the DRC is to gather the data and the metadata. Now, one could always go to the study itself and find these things, but much of it is in context really of the study itself and not overall across the Common Fund programs, which makes searching and retrieving that information a little bit more difficult. But all that data is there, and this is how we are able to take the data from any one of our Common Fund programs and bring that information into it. Next slide. And here you can see a lot of the data types and information that we've incorporated into C2M2, but only because Glygen was there to actually give us a lot of that information. So, for example, there's a proteome master list. And if you look to the left, if you were just interested in the hepatitis C virus, you can collect that kind of data. And again, the DRC and the C2M2 model allows you to then go across various programs to get specific information that may be related or may be categorical for that. So you can put that data in context and work across. Next slide. So as we've talked about really, how we integrate the metadata and point to where it all is. The next part is what do we do with that information? And so this right half really shows how we're able to use some tools and use cases to take that data and work across the data and resources across the various DCCs to actually make things that are entirely usable by you just through exploring the DRC. So let's get some examples on the next slide. So one of the things that we really like at the DRC is the gene set card, and it's a way of enabling you to fetch gene sets from Common Fund programs. And one of the things to remember about the various programs we have is what is a common element that we can integrate across? Well, big thing for us is biological mechanisms, and underpinning many of those are genes. And so this is why the power of gene sets, and I know Noel's going to talk about that a little bit later, but that's one of the really hallmark things that we're using because genes go across our various programs. And I'm not just saying this as a geneticist, but it really is a great way between genetics and the transcriptome of allowing you to go across the data and harmonize and really kind of anchor a lot of those fundamental observations. And that infrastructure for that is provided by the DRC. Next slide, thanks. And really, you could see the large list of tools and workflows going across. And this is also provided within the DRC. So if you want to get more information, and a lot of the things that we generate are annotated and stored. We have references here. We point you to the GitHub, so you can actually take these resources and use them. Next slide, we'll talk about one of them in a moment. So the Playbook Workflow Builder, we're going to talk about two aspects of it. This is a really cool way. It allows you a relatively easy way to point and click your way across our data sets and do a series of computes to actually get a published workflow that goes across Common Fund data sets. So this is something that anybody can integrate into their own workspaces, into their own programs across the country. We're making this available and allows you to really bring the resources of the Common Fund and this methodology of creating clickable workflows to resources everywhere. So it's integrating the DRC, and we're integrating it across. For example, it's going to be available in our cloud workspace, which Noel will be talking about later. So this is one of the really great things that we're excited about. And we have use cases and workflows to show you how to use it and actually make discoveries for yourself. Next slide. So one of the things that we wanted to say, within CFD, we had one of the first chatbots at NIH, and we're really excited to do that to help you understand how to, in a textual way, how to find out about data, resources, and even provide some visualization of what the data looks like for anyone who's interested in our data sets. So that's provided by the Playbook Workflow Builder. But it also has a really nice feature that it will automatically generate method text with references for the workflows that you build for your publication. And one of the things that we'll be adding soon, making available through Playbook Workflow, one of its features, is actually some visualization tools that you can use to help you actually take your analyses and make things that you can publish with. So that's something that's underway, and that's also really exciting. Again, we want to help you get that information and help you even in presenting some of that information for publication. Next slide. This is an example, and I think we're going to try to share it with the group later of, and I'll try to find and put in the links, is an example using very disparate DCCs, but really integrating across. And it was published in PLOS Computational Biology, really showing you the strength of this and how you can it's a way of showing you how you could actually do this yourself and start processing your own data or using common fund data itself. So it's a really nice example. Next slide. And here are examples. Oh, I think we want to go to GeneSet. There we go. And so this is GeneSet Cart, which Avi has been working on and is presented out there. And it's a really nice way of actually allowing you to create your own gene sets. And again, Noel will be talking about that, but it's a very easy methodology for doing that. So it's a great thing, and I want to make sure people are aware of it. Next slide. And you can also, using gene set crossing, we can go across common fund programs and add overlapping genes to a cart and do these analysis and bring them in for analysis. So that's another way of really going across these various studies. And you can see, for an example, here you have 8 different studies going across. Next slide. And now I turn you over to Noelle, who will be building on some of the things that I mentioned, and she'll be talking about the CFD Knowledge Center, which she knows very well. Thanks for your slides, Noelle.
No problem. All right. So George gave you a great landscape of the DRC. I'm going to talk about the Knowledge Center, which focuses on a different axis of the Common Fund, which is about the knowledge and the results. And it's sort of, we are the yin to the yang, if you will, of the DRC. So we're trying to improve understanding and visibility across the CFD resources. We really want to aim in being a centralized entry point for understanding the knowledge across the CFD. We are disease focused and we're integrative with the goal of really driving discovery. So how do we do that? We do that by a concept of really defining knowledge in 4 terms. One is providing foundational knowledge across all the CFDs. What is the purpose of the program, right? Primary knowledge is the second type of knowledge, published findings. What has each DCC learned from their expertise over the 10 years and all the data they've aggregated? That's a lot, but we try and focus on that in very specific ways. Secondary knowledge. These are things that can be obtained from a single DCC, but not necessarily published or done. Like where the DCC is like, oh, it would have been really nice if we could have analyzed this for disease status, because it wasn't necessarily their remit. We would work with them to do that as a partnership with them and make that accessible in the Knowledge Center. Or tertiary, which is really the exciting stuff. These are assertions created by integrating data across multiple programs, creating new analyses, and really the full enchilada here is really thinking about how to work with other resources. And I'm going to talk about a couple of the key programs that we've done that for in a minute. To really accentuate what I mentioned earlier about the key thing about CFD, which is the resource of resources. And if it does it really well, it actually really enables other resources to rapidly contribute and collaborate with us. So with that, here's a CFD Knowledge Center, and just gives you a place. This is really like a navigational infograph where you can navigate across the different programs and learn about them. So for example, if you highlight MotorPack, you'll learn that like what omics data it has, what entities it indexes on. What we've also done is try to give you foundational knowledge. So with the DCCs in that community, we've curated summaries of all the DCCs in terms of like primary and foundational knowledge, not necessarily where all the data. The DRC does that beautifully. We think about the knowledge, what you can learn. So this is done for all of the DCCs. And again, this is interconnected to the DRC. So if you just want to go from there down to the DRC to get the C2M2 model data or any other data from the MotorPack, for example, you can do that through here. Or if you want to go to MotorPack directly, you can. So it's all connected, right? So this is learning at a high level. Next level down is really like we wanted to bring at one place primary knowledge generated by these DCCs that for a given entity like a gene, a disease, a cell type, a biological process, you could actually just learn what they know. So here's some examples of some primary knowledge from IDG, where it's basically if you type in your favorite gene, it tells you what they believe is the drugability of that particular target. Or from Glygen, find out right away if your gene that encodes your protein, its glycosylation points and how it's glycosylated, and you can link directly to Glygen from there. That gives you the sense of primary analyses. Secondary is really fun. This is where we've worked with the DCCs, where they've been like, hey, we've always wanted to do this, but we haven't been able. So here's an example we did with Metabolomics Workbench, where we took all of their metabolite data for common disease. We, we structured it consistently with their methodology, what they recommend in Metabolomics Workbench. We compared to all of our human genetics data we have in another resource, which I'll talk about in a minute. So therefore now on the Knowledge Center, you can say what metabolites are associated with your favorite disease of interest using human genetics as that backbone. So it has like actual data and analysis from Metabolomics Workbench that couldn't be done anywhere else, that is only enabled because of that unique data set that they have, right? So it's all those tools and principles taken into another analysis, hence the secondary analysis. Here's another example of secondary analysis where, HubMap, if you're really an expert in HubMap, you want to go to HubMap. But if you really want to just know for a given cell type or tissue or gene, what it looks like across single cell data from HubMap, we've created an easy way for you to do that. So we've created integrated maps with the help of HubMap from all of their tissues and integrate them in one place so you can navigate them and see gene expression data on the fly. What's great about this, again, it's an entree, it's the real data, it's curated with them, but it's done in the context of all the other information that we have on a given tissue, a gene, a process in the Knowledge Center. Again, these are results and knowledge all bubbled up to you from the data coordinating centers and the DRC level. Here's another example for Kids First. This has been actually a really fun collaboration. These results are not publicly accessible anywhere but on the Knowledge Center, which is great because Kids First has very protected data, right? But what we've done in partnership with them is run their protocol and done associations to figure out how you can done association of variant counts and consequences for all their rare pediatric disorders. So what's great about this is this serves as a nice validation. If you have a gene that you're working on for a particular phenotype, you can look up what Kids First says about it and all the consequences. We know this is valid because it produces the examples from the literature. For example, I gave you one here with cleft palate. This is observed only in Kids First, but not in NOMAD, for example. So NOMAD is another resource that you would probably go to if you have a variant of interest and you want to see, its frequency across many, many populations across the world. But Kids First tells you exactly what they saw, about cleft palate. So again, this is in one place, all brought together. And the next is, sort of what we really want to do, right? So we really want to say like there's secondary, there's primary, there's all these things together, but to really drive discovery, you need something that is basically transformative, right? Or translational. And I think tertiary analysis starts to get us there. So what really want to make us possible is work that spans DCCs and centers and serves as a substrate for external connection. So a way to do this is with something George alluded to, which genes and gene lists. This is essentially a universal language. It's like a commodity across CFD. It is great because all of the CFD programs are actually informing on biological mechanisms in general, right? Genes are simple, concrete, easy to generate lists of genes, right? They're easy to generate because they can come from, they're small, right? We have infrastructure as the DRC has built to capture them, analyze them, and generate them. So obviously done a lot of work to create all these gene lists from all these DCCs. But what's great about them is they provide biological mechanisms can be partially captured with gene sets, and they can help us understand important mechanisms and hopefully give us impact on human health. So to that end, what we've done is created a resource within the Knowledge Center to link gene sets or lists to both common and rare human and mouse phenotypes. And we think this is very valuable because it really takes some commodity across all of our CFD programs and makes it elevated to a broader user base, but also relevance to human and common rare diseases. So what's great about that is we had to apply a statistical framework. This is work done by my other principal investigator on the Knowledge Center, Jason Flanick. This is a method that basically goes beyond classic enrichment analyses that are done in genetic studies. You may have heard of MAGMA. This is another one called Pigeon. It's currently unpublished and we're working on a publication right now for it. But what's different about this is it really uses human genetics as a backbone. And so you can look at this method. We also have a webinar I can share with you about this method. I'm not going to focus on the method too much, other to describe that it allows us to connect gene lists through annotations and through a probabilistic model to human phenotype and both mouse. So here's what goes into the model, right? We have full summary statistics from the A2FKP, which is a NHGRI funded resource where we've taken curated summary statistics across 1,300 traits and integrated in the resource. Again, we also have pulled in all of the data from GWAS Catalog through a collaboration we have with GWAS Catalog into this resource. Then we also have brought in OrphaNet data to complement with rare disease. Then we've run differential expression through all of the CFD data sets that have that and applied them across these gene sets that Avi's group has created. And this all produces essentially a substrate for collaboration because it takes the information in these gene sets, links in the human phenotype to give proposed and possible mechanisms, at least a place to jump off, like a hypothesis to generate. So under that model, where you find this on our landing page. And so I just want to give you a quick vignette, and then I think I'm short on time, but this will be useful for you, I hope. So MotorPack is a great example, right? So they generate a lot of omics data across human and rat, but they're doing this specifically for exercise and chronic disease and applications of chronic disease. And they create a lot of these results, right? But wouldn't you want to know how that relevance in rat is relevant to human? So one of the things we've done here with these gene sets that were enabled through the DRC, through the work MotorPack did, is you can type in your favorite phenotype. And I use the example of red blood cell count here, and you'll see what genes are associated, what gene sets are associated with that phenotype and what gene set that is and where it comes from in MotorPack. For here, this is the second to highest result we have in this particular, for this phenotype. And these are the 184 genes here, and you can explore these genes and see what they're involved in. But moreover, what I like about this is we also provide statistical inference based on genetic support, and we define all these things for you so you can understand, where we got this information. But also more importantly, there's essentially, some support to some of this work, and I'll just give you a sense of something that we think is validated in the sense that what we found here is supported by the literature already within a study where, people who have sedentary lifestyles, actually with exercise, their hematological industries improved. So there's evidence that red blood cell count also increases in humans, as we saw it also in the MotorPack upregulation in blood cell count in the rats. So there's just like validation there, but this gives you the idea of the type of things that can be discovered using this module. We've done the same thing for humans and to mouse, which is really useful because a lot of clinical studies are done in mice. And how this is typically done is you observe something in a mouse, and then through semantic similarity, you assume the human phenotype. Well, we want to use genetics for that because we're hoping that this will make it better for those preclinical models, because often the mouse phenotype does not necessarily recapitulate the human phenotype. So for example, here's an example from head bobbing that I use. So that's an observation in a mouse. And this is what, the semantic resources say. And that's pretty good. It gives some biological things with neurological function. So that is good. And so these are really useful, but couldn't you add even more information to this? So we've done this with the resource as well through all of our mouse gene sets. And what we've been able to produce is a result that gives you a sense of the things that are genetically correlated with a phenotype in mouse to humans. So for example, if you typed in head bobbing, here what you get is rare deafness. So obviously this is like not necessarily logical, but you can imagine a mouse, when they go to here, they bob their head to try and get a sense of what's going on around them. And that actually could make sense, but you would never know that normally unless you use semantic search, or in this case, genetics, because it gives you even more support for what that is going on for the human phenotype, for example. So this provides insight. Hopefully this is better targets. We have a lot of work to do here because it's new. And we also want to work with human mouse geneticists to actually understand this. We want to get this in front of them because they're really the best folks to validate this. So this just gives you some examples of what's happening in the Knowledge Center to take all of the work that's been done by the DCCs and the DRC, bringing it together and creating a commodity that hopefully has relevance also to human phenotypes. So at the end, I'm just going to get through this. Actually, do I have a couple more minutes? Let me know if you want me to stop, but I'll go a couple more minutes just to wind up and talk a little bit about
2 minutes if you don't mind. Yeah, you can.
I can do that. I can do that. So a couple other centers that you're not going to hear from today is our Cloud Infrastructure Center, funded at TAC, and they are a team built to bring all of the CFDA data into a cloud environment so that you can work with it. What's great about Galaxy is it's open source, it's free, it's been around forever, and these are a great group of people, and you'll be hearing more from them as a place for you to have a workspace to do your own analyses and compute. There's already tools and interfaces they've already built, so that's fantastic. Keeping us all coordinating is Jake and his team at the Coordination Integration Center. They have 3 things that they're responsible for, but they're responsible for so many other things, like keeping us all organized. They do our sustainability, our administration, and evaluation. They make sure we all run smoothly. And then you also heard about the training center. But what I want to mention about the training center is what's great is that they're doing these wonderful hackathons. And I've never participated in these until recently. But what's really cool about this is it's an example of using data across CFD resources. And this is the type of work we want to see more of. So hopefully you all are inspired today by that. So in the future, we have a lot of work to do for CFD. We are still, in the early stages, as George mentioned, but hopefully our success will be measured by you all using what we've built and enabling you to make better discoveries or connecting them to other tools. We want to enable biologists. We want to enable computationalists. We want to create more killer apps. But we really want to innovate. And lastly, I think networking with external resources really our goal. There are so many NIH resources out there that have similar structures. They have small data resource centers within them that also are trying to come together for a specific disease or purpose. And so our hope is that we can work together with those other NIH resources to create an even wider ecosystem of resources through the model. This is the inhabitants of our ecosystem. I want to thank all of them. They're wonderful people, and you're hearing about all of their work today, so I can't represent all of them. These are some of the wonderful people I get to work with every day. And with that, I will stop and hand it back over to the training center. Thank you for your time.
Thank you, Noelle and George, for this wonderful presentation. As you can all see, there is a lot that is part of the Common Fund data ecosystem. So if you're sitting here thinking like, wow, so many tools, so many resources, so many ways to get involved, maybe you're thinking about the students you have, or you are a student and you're wondering, okay, I need even 3 more steps back on how to kind of engage with the ecosystem. I'd like to introduce my colleague, LaFrancis Gibson. She's going to talk about building bridges and how the CFD Training Center is helping power that discovery, and particularly, hopefully, helping make even more of those kind of connections in ways where you can more easily break into this ecosystem. So LaFrancis is our contact PI for the CFD Training Center and manager for health promotion at ORAU. She has more than 15 years of experience in managing outreach initiatives, training development, and program evaluation, and has worked across a lot of government agencies in different contracting roles, including NIH, National Library of Medicine, and the Centers for Disease Control and Prevention. So LaFrancis, thank you, and go for it.
Thank you so much. So good afternoon, everyone. And I just want to quickly say thank you, George and Noelle, for the great presentation that you provided on CFDE. I think it makes a great transition for me to talk about the CFDE Training Center and what we're doing to further promote CFDE. So I'm excited to spend the next several minutes just discussing how the CFDE Training Center actually powers discovery through the many training resources we offer. Next slide, please. All right, thank you. So before I jump in, I do want to spend a few minutes just providing a brief overview of the CFDE Training Center. And so we were actually funded September 2024, so we are still relatively new, but our overall goal is to act as a central hub for training, providing a comprehensive approach to support current and potential CFDE users on their learning journey. Our focus is on providing training in basic and advanced computational and data analytics skills for those interested in CFDE data, tools, and resources. Our objectives include coordinating with other CFDE centers and funded programs and projects to meet training needs and to expand the CFDE data user base and enhance the confidence and complexity of data usage through community building and engagement activities. To support current and potential users, the Training Center has created a series of activities, including, as you see here on the slide, a podcast series, as well as an e-learning dashboard for training, also hackathons and workshops, a mentoring program, and meeting forums such as this symposium. Next slide, please. So to reach our goals, we first had to take stock of where things stood both within CFDE and the broader training space. So our team actually conducted a landscape analysis between November 2024 and February 2025 to determine what's working, what's missing, and where we can improve so that we could make well-informed decisions moving forward. Next slide. And so the landscape analysis we conducted actually turned out to have a mixed method approach. And so what we ended up doing was reviewing existing CFDE trainings, also conducted key informant interviews with CFDE subject matter experts and learners. Also, a systematic literature review was conducted, as well as CFDE training survey. A mentor survey went to our ORU consortium. We also took a look at academic engagement with CFDE datasets, and then did some information gathering from the data coordinating centers. Next slide. And so the key findings that we found from the landscape analysis, there were several gaps that needed to be addressed, as well as expanded to actually meet user engagement. So first, we heard about learners feeling like they were overwhelmed by the number of platforms and places for information within CFDE, with unclear starting points for beginners. And so they also wanted more diverse and interactive learning methods with smaller bite-sized modules they could explore during their short downtimes. As far as summer programs, some of the barriers we uncovered were mentee preparedness and mentor availability frequently came up. Also, we learned that trainers themselves sometimes found it very challenging to maintain learner engagement, to assess progress, and to assure that the training translate into real-world application. And so there were also some struggles we identified with some of the training material and just having that assessment standardization across programs and platforms. Learners also expressed the need for continued support via initial training sessions and more feedback mechanisms to understand their progress, whether they were a beginner to expert level or intermediate. And so how exactly did we, the Training Center, bridge those gaps? Next slide. So to address some of the issues surrounding complexity for beginners, we actually created a how-to seminar series that provides an introduction to various omics and omics-related tools and techniques while highlighting the programs within CFDE that utilize those data sets. So in our how-to series, we have been able to feature data and tools from GTEx, XRNA, HubMap, Kids First, 4D Nucleum, Links, MotorPack, SendNet, and Spark, just to name a few. And the how-to series is currently available on our e-learning dashboard, which I will share more information about a little later in the presentation. Next slide, please. Okay, and so we also developed our Decoding the Data Ecosystem podcast. This is a series that's dedicated to having casual conversations about the CFDE infrastructure, as well as complex omics ideas. We already have 8 episodes available with topics ranging from the CFDE knowledge and training centers, reproducibility utilizing the CFDE Kids First data, as well as exploring FAIR principles to the hackathon at the Bio IT World. We have a series on the knowledge graphs, as well as the landscape analysis and the importance of public data sets in cloud computing and scientific research. You can find these podcasts in most places. You currently enjoy your podcast, so we are available on iHeartRadio, Apple Podcasts, Amazon Music, and Spotify, just to name a few. Next slide. Thank you. So next, to address what we heard with the mentorship challenges, we created the Decoding the Data Ecosystem Collaborative. This was actually established to foster collaboration and engagement amongst the training center and a broader research community. As part of this effort, we did establish our mentoring program, which actually utilizes a comprehensive mentorship system involving a technical mentor from a data coordinating center in the CFDE. And these mentors actually deeply understand the data and the tools for the scientific question posed by the mentee team, which comprises both a faculty PI and a student. And so we actually just completed our pilot mentor program. It was a 10-week program that included a series of activities and events. Next slide. And so the goal of the mentoring program is for faculty PI to incorporate more CFDE resources into other activities such as classes taught and students mentored. In addition to that, the student benefit is to receive support for a summer research with multiple mentors. The CFDE datasets utilized in the projects this summer included HubMap, IHMP, GTEx, and 4D Nucleum, which were ideal for student researchers as these datasets offer public available summary information and not just controlled data. Mentee teams also utilized non-CFDE datasets and tools, including those such as whole genome sequencing, WGS data, CM4AI, and eQTL reference data, to name a few. This just reflects the ability to utilize a combination of datasets and tools with the CFDE datasets for research. The pilot program, as you can see, actually included 4 mentee teams. And so we will hear from them a little later in the presentation as they will showcase their research projects for us. Next slide. And so moving into resource implementation, we actually created an e-learning dashboard. And so this is where we house our how-to series mentioned previously. And also the really great feature about the dashboard is that it organizes content into the type and difficulty level. We are currently working on building out our training content by partnering with other CFDE programs and centers to ingest their most popular training materials, as well as plans for supporting training modules built in partnership with the broader scientific community. As we add more content, we will have the ability to build out the learning paths as well. Next slide, please. And so now I'm actually really thrilled to share this summer we actually recently launched our CFDE Training Center website to serve as a central hub for connecting to our trainer resources. So I will switch gears and just demo our website. If you give me just a second. Okay, so hopefully, can everyone see everyone can see my website. So just want to kind of talk through a few key features of the website. Here on our homepage, we do feature our upcoming events that are happening, as you can see with today's event. Also, we provide really quick access to connect to our Learn, Connect, and community resources as well. Also from the homepage, you can access registering for our online e-learning dashboard, as well as checking out our podcast series. And then we always try to feature some of our most recent activities that have just taken place within our activity schedule. And so here at the very top, we do have quick navigation to allow you to connect to some of our mini sections. So our About section, if you are interested in learning more about CFDE and the training center team that is putting on a symposium, you can definitely connect with us here. Also, we have our Connect section, which features today's symposium, as well as the podcast and any upcoming events. And so specifically with the podcast, I do want to kind of shout out that this is a really good resource. Again, if you are looking to get some valuable insights, here's some very inspiring stories about what's happening in the Omnics research space. And this is hosted by our training and engagement director, Dr. Alyssa Dillman. And so also, as far as our upcoming events, if you're ever curious, what is the training center doing? Where is the training center going next? We do feature that here. But another really cool resource, if for some reason you can't make our event, we do also feature our previous event, so you can always stay connected with what we have going on. Also, at the very top navigation, we have our Learn section, and this connects you directly to our e-learning dashboard. Again, this is a free resource, really simple registration process that you can sign in to get started on connecting to the trainings that's relevant to the CFDE universe. And then we have our collaborative section here as well, which at the moment, of course, we're featuring the mentor program that we just completed. And then lastly, I do want to just point to the very top of the page where we do directly link to the information portal and the data portal, knowledge portal, and coordination portal that was shared in an earlier presentation. So if you give me just a second, I will switch back to my slides. All right. So moving on into our engagement implementation. So finally, I do want to highlight, and I know Noelle kind of shouted this out earlier, but we had our fair and open source hackathon we ran in April at the Bio IT Worlds. We did have 5 teams in total that were participating. And so the data sets that were utilized, you can see here in this image, MotoPack, IDG, GTEx, and HubMap. And we also provided a link here to the GitHub repository. So if you would love to like explore more, the link is available. Next slide. And so more on our engagement and implementation. So most recently, the Training Center and Engagement Director, Alyssa, actually recently presented at the joint ISMB and ECCB conference in Liverpool. Alyssa's presentation was centered around building omics skills through the CFDE Training Center. And so this was a really great opportunity as it allowed for the Training Center to have more of an international reach while promoting the CFDE and Training Center resources. Next slide, please. So what's next? So as far as what's next, what's coming up really, really soon, the Knowledge Center is actually hosting an after-hours event at the Westin Boston Seaport District on Tuesday, October 14th from 7 p.m. to 9 p.m. This is actually open to anyone interested in learning more about CFDE. So if you're local to Boston or be in the area for the ASHG happening that week, feel free to drop in to meet some of the researchers from across the CFDE network and to learn more about the resources and tools. Included here is a QR code for registration. Next slide. And so also at ASHG, the Training Center will have a poster presentation that is taking place on Friday, October 17th. We also plan to host another hackathon at the Bio IT World in 2026. It is tentatively planned for May 18th through the 19th. I will note the hackathon typically takes place one day prior to the conference and all day on the conference start date. So in addition to that, we do have some planned activities for FY26, which includes an expansion of trainings to modular in-person workshops at academic universities. And so what we're planning for are 2-day skill building workshops for both undergraduate and graduate students. And so if you have an interest in us possibly hosting at your university, please feel free to send us an email. Also, we do plan to expand our asynchronous material in e-learning dashboard. Also, expansion of our podcast and podcast guests. We also plan to expand the collaborative. So look, coming out fall 2025, we do plan to launch the application for our next mentoring program cohort. We've had some really good feedback from this pilot program, and so we're actually looking to incorporate that and just further refine the mentoring program. And so if you're interested, definitely please be on the lookout. Also, please come back and check our website out as we will be posting information once we're ready to release the application. Next, we do plan to enhance CFDE-wide training and engagement collaborations and opportunities while also hosting quarterly open house office hours. And so with the open house, we do plan to have more of a focus on some of those technical questions that that typically arise within the CFDE datasets. And then lastly, we do plan to continue our ongoing evaluation and curriculum development. Next slide. So thank you all again for for first being present for the symposium today, but then also allowing me the opportunity to tell you more about the CFDE Training Center. I hope you learned a lot about what we're doing and the direction that we're headed. If you would like to learn more about CFDE, I included a link here, as well as learning more about CFDE Training Center also. If you're interested in collaborating, getting together to be a podcast guest, feel free to shoot us an email. We are more than open to collaboration. So with that, thank you.
Thank you, LaFrancis. And with all of these presentations, I hope you're starting to get a sense of the Common Fund data ecosystem and all of the opportunities and data sets available. All right, so we're to our Mentee Showcase. This summer from June to August, the CFD Training Center launched a pilot mentoring program. LaFrancis talked a little bit about this as well during her presentation. The program really aimed to support professional development through pairing mentors from our CFD data coordinating centers with our mentee teams made up of a faculty PI and a student. The program aims to provide both faculty and students an opportunity for direct engagement with CFD program opportunities, and also more in-depth integration of CFD data within the academic learning environment. So our next session, the Mentee Showcase, celebrates the achievements of our Summer 2025 Mentor Program participants with presentations highlighting their journey, projects, and the impact of mentorship on their professional development. So first I want to welcome Molly Huang, presenting on her multimodal analysis of DNA repair genes in the placenta. Molly is a bioinformatics and systems biology PhD student at UC San Diego, advised by Kathleen Fish. She was mentored this summer by Kathleen Fish of UC San Diego and Ho-Han Wang with the CFD GTEX program. Welcome, Molly.
Thanks for the introduction. So hi, everyone. I'm going to be presenting on my presentation. And like Jennifer mentioned, I am a third-year bioinformatics PhD student at UCSD. Next slide, please. So for this talk, I'm going to briefly go over what the placenta is, and my first task for this project, which is looking at DNA repair genes in placental trophoblasts. My second task for this project, which is integrating placental gene expression with GTEX, and then some conclusions from this work. Next slide, please. So about the placenta. So the placenta is a temporary organ that develops in the uterus during pregnancy. The purpose of the placenta is to provide oxygen and other nutrients to support fetal growth, as well as remove waste and carbon dioxide from the fetus. So while the placenta is crucial to fetal development, it can also be detrimental to maternal fetal health. And this is because it's involved in a couple of different diseases, which include preeclampsia, which is hypertension that occurs during pregnancy, as well as maternal vascular malperfusion, where lesions develop on the placenta that impair its function. So this is why it's really crucial to study and understand both the healthy and diseased placenta. Next slide, please. So for my project, I'm really focusing on the placenta from the DNA repair gene expression angle. So for the goal of this project, I want to understand how expression of DNA repair genes differ between placental trophoblast subtypes. Placental trophoblasts are the main cell type of the placenta, which I'll talk a little more about, as well as between the placenta and other organs. Next slide, please. So one part of this project, in terms of background, that I think is important to understand is DNA repair. So for this project, we use Reactome, which is a CFDE tool, to capture the DNA repair pathway, which is able to provide a list of genes that are involved with various DNA repair mechanisms. So the goal of DNA repair is to remove or mend any DNA damage that can arise from things like environmental exposures or errors that occur during DNA replication. And this can be done using several pathways that are shown in blue in this diagram from Reactome. So one pathway could be base excision repair, which is shown on the top left, and this repairs any damaged bases, or there could be mismatch repair, which is shown on the bottom right, to repair any base repair mismatches. And this is among many other pathways that are captured in Reactome. Next slide, please. I think another portion of background that's important to understand for this presentation is what trophoblasts are. So trophoblasts are the main cell type of placenta. They arise from the outer layer of the blastocyst, which is the 32 cells around the 32 cell stage. So the outer layer of the blastocyst is made of trophoblast stem cells. These then derive into, next please. Cytotrophoblasts and, extravillous trophoblasts and syncytiotrophoblasts. Next. So if you look at the cell level of the placental vasculature, you can get a good idea of the function of these trophoblasts. So you can see on the placental villus structure, which is at the top of the diagram, in the interior, you can see that there are cytotrophoblasts. This allows it to then give rise to syncytiotrophoblasts as well as extravillous trophoblasts. You can see that the syncytiotrophoblasts are on the exterior of the placental villus structure. This allows it to facilitate nutrient transfer between the fetus and the mother. And then the extravillous trophoblasts are located in the middle of the diagram between the placental villus structure and the decidua. This allows it to facilitate spiral artery remodeling. So you can see on the bottom of the diagram, the blood vessel is increasing, and this allows for increased blood flow and nutrients into the placenta. So this kind of shows how the trophoblasts are really necessary to placental function and therefore necessary to understand. Next slide, please. So one interesting feature of trophoblasts that have been found is that they have a really high mutational burden. So you can see on the y-axis, this paper plotted the coding substitutions, so the amount of mutations per megabase per year. And you can see along the x-axis, they plot a bunch of different childhood cancers, and this includes like leukemias as well as other cancers. And the rate at which trophoblast clusters are mutated are very similar to these childhood cancers, and childhood cancers are known to be very highly mutated. So this kind of made us really interested in how because of the high mutational burden, this relates to like DNA repair during trophoblast differentiation. Next slide, please. So previous work in my lab has looked at DNA repair in trophoblast cell lines. So we took induced pluripotent stem cells that were then derived into trophoblast stem cells, which kind of represent like progenitor cells, and then we derived them into extravillous trophoblasts. So when you look at normotensive extravillous trophoblasts versus normotensive trophoblast stem cells, you don't see any dysregulation of DNA repair. However, when you incorporate preeclampsia and MVM, so diseased extravillous trophoblasts, as well as diseased trophoblast stem cells, you start to see this dysregulation of DNA repair. So this kind of shows how DNA repair is relevant to healthy differentiation of extravillous trophoblasts. And interestingly, you also see dysregulation as well as just expression of different pathways that are relevant to DNA repair, such as the P53 pathway, as well as the G2M checkpoint. So this kind of shows like why it would be interesting to study DNA repair in the placenta. Next slide, please. So because that previous work was done in trophoblast cell lines, we wanted to expand this into the actual placenta. So one data set that we thought to leverage was the HubMap Female Reproductive Tissue Mapping Center. So this mapping center is focused on understanding the cellular composition and architecture of the placenta. And it does so using these various molecular assays. And I will go more into depth about the specific assays that we use. Next slide, please. So with this data, we kind of move on to task 1, which is looking at DNA repair genes in placental trophoblasts. Next slide, please. So from HubMap, we got 11 total samples. These samples came from a variety of sites. We looked at site A on the placenta and site B on the placenta, as well as the fetal region, which is the region of placenta closest to the fetus, and the maternal region, the region that's closest to the mother. So this allows us to get a variety of locations from the placenta. And these samples had balanced fetal sex as well as delivery method, and they were all around 39 weeks of delivery, meaning that these were term placentas. Okay. So we got single nuclei multiome data from these term placentas, and we get 9 general cell types from these 11 different samples. So these cell types range from endothelial as well as fibroblast cells to various immune cells to the trophoblasts. So you can see on the right of the UMAP plot, the majority of these cells are trophoblasts. You can see like that giant like purple cluster. And this makes sense because it's like the main cell type of the placenta. So this is the data that I'll be using for the rest of this task. Next slide, please. So in order to look at DNA repair genes in placental trophoblasts, we got the HubMap single nuclear multiome data. And then we processed them using a tool called Seurat. And then we did pathway enrichment scoring for DNA repair genes using a tool called UCell, which I'll explain more in the next slide. And then lastly, we used Seurat to look at differential expression in Slingshot and TradeSeq to visualize trajectory in order to pull out individual expression of DNA repair genes. So the first thing that we did was we looked at gene signature scoring. So what gene signature scoring does is it quantifies the activity of genes in a set that's given with the goal to characterize like different cell types, cell states, as well as the active biological processes or responses to environmental cue within those cell types or states. And we did so using a package called UCell. Please. So for our samples, we looked at the reactome DNA repair signature to see which cell type has the highest reactome DNA repair activity. And we see that the trophoblasts have the highest DNA repair gene signature. It's higher than every single cell type except for the macrophages and lymphocytes. And this is to be expected considering that the previous paper had shown like the high mutational burden of the trophoblasts compared. So it should be higher than the other cell types. Next slide, please. And then we wanted to look into the trophoblast subtypes to see which subtype has the highest DNA repair gene signature. And we saw that in the cell types that the cytotrophoblasts, which are the progenitor cells, have the highest DNA repair signature. So this is plotted along the course of differentiation. So the cytotrophoblasts are higher than the syncytiotrophoblasts as well as the extravillous trophoblasts that it differentiates into. Next slide, please. And next, we wanted to look at individual gene expression of DNA repair genes. And in order to do that, we needed to be able to visualize trajectory. So one tool that we used to visualize differentiation trajectory was called Slingshot. You can see in this plot that it goes from the cytotrophoblasts in blue to the proliferating cytotrophoblasts in gray, and then the early syncytiotrophoblasts in pink, and then lastly, syncytiotrophoblasts in purple. So then we'll use this plot to look at the individual gene expression. Next slide, please. So yeah, this builds out the cytotrophoblast to syncytiotrophoblasts differentiation. Next slide. So we are able to capture from this DNA repair genes that are temporally dynamic. You can see in this plot, there's a gene called NSD2, and it is more highly expressed in the cytotrophoblasts. And then in the literature, we found that loss of function mutations in NSD2 is associated with abnormal placentation in mice. So this kind of shows how it's both a DNA repair gene and relevant to placenta. And then we can also look at TRIM25, which is more highly expressed in syncytiotrophoblasts. And TRIM25 in the literature has been shown to be essential for estrogen-dependent cell proliferation. So from this, we can pull out DNA repair genes that are both temporally dynamic and relevant to the placenta. So this kind of shows how DNA repair genes may be relevant to trophoblast differentiation. Next slide, please. So for the second task of this project, I wanted to kind of expand outside of placenta and look at the placenta in comparison to other organs, particularly in DNA repair genes. So in order to do that, we integrated placental gene expression with GTEx. Next slide, please. So, sorry, next slide too. So what GTEx is a comprehensive public resource to study human gene expression. This is a CFDE program. And it looks at 54 tissue sites across nearly 1,000 disease individuals. So there are various organs that are covered, and then there are some organs, such as the brain, that are divided into specific regions. So this allows you to really look at gene expression changes between different tissue sites. Next slide, please. However, the GTEx dataset does not look at placenta. So one of the goals for my project was to integrate the HubMap placenta dataset. Next slide, please. So within the HubMap placenta dataset, we had bulk RNA sequencing from 12 different donors. We looked at 6 samples per donor. So we looked at the site A and site B that I mentioned earlier, as well as the fetal, maternal, and central region. So this allowed us to have 72 samples in total. Next slide, please. So in order to integrate placental gene expression with GTEx, we followed the methodology that was done previously in GTEx. So we had the HubMap bulk RNA-seq BAM files. These were then mapped back to FASTQs using GATK. And then they were aligned using STAR. And then we used RSEM for gene quantification. And then we integrated in the transcripts per million measurements from other tissues that were provided in GTEx, and then did all the visualizations in R. And then to note, the transcripts per million values do not utilize batch correction for covariates in GTEx. So we're able to replicate the GTEx bulk tissue expression with placenta. So one of the genes that we looked at earlier, which was NSD2, that was temporally dynamic, you can see all of the GTEx tissues plotted along the x-axis with placenta, and you can see that the NSD2 expression is very similar in the placenta to other genes. Next slide, please. Or sorry, other organs. But with TRIM25, you can see the expression of TRIM25 in the placenta is a lot higher than the other organs. It's very similar to like the whole blood level, which is another like tissue that has high somatic mutational burden. So this just kind of just describes like the function of the integration, which allows us to compare like the expression of individual genes in the placenta with other organs in GTEx. Slide, please. So another thing that I wanted to look at was whether placental region contributes to changes in expression, because in some tissue sites in GTEx, they do divide it into different regions. So I looked at reactome DNA repair genes that have been expressed in the placenta, and you can see that there's no change between the maternal, central, or fetal region. So we decided to proceed with the placenta regions all combined into one. Next slide, please. So next we wanted to look at if there was tissue-specific DNA repair gene expression. So we plotted out all of the reactome DNA repair genes, and we're able to see like 2 clusters. There's one cluster on the right, which has high DNA repair activity, and then there's one cluster on the left, which has low DNA repair activity. And this is the cluster with placenta in it, which is boxed in purple. But interestingly, it has this like red block of highly expressed genes. So I want to explore what this red block was further. And then interestingly, this red block of highly expressed genes are all histone genes in the reactome DNA repair pathway. So you can see them listed out on the right. Next slide, please. And these are also highly expressed in tissues like the prostate and the whole blood, which are found on the right, except it's obscured by the high placenta signal. Next slide, please. So this kind of shows how, because histone genes have such a higher expression in placenta, they're potentially an avenue of exploration. This has previously been looked in other literature, such as in this previous paper that looks at histone modifications along trophoblast differentiation, as well as just epigenetics in general and how they relate to different pregnancy disorders. So I think that this shows that from this GTEx integration, we can look at, explore, we have future avenues of exploration. Next slide, please. So some conclusions from this work. We've found that trophoblasts have a high DNA repair gene signature, particularly in the progenitor side of trophoblasts. Additionally, temporally dynamic DNA repair genes relevance to trophoblasts can be a future point of interest, like NSD2. And lastly, we integrated placental gene expression with GTEx, and this allows us to compare individual genes in the placenta with GTEx, which can be a tool for placental biologists. Next slide, please. So some challenges from this project. I definitely had a lot of issues with converting between different languages and data types since we were aggregating a lot of different data as well as just using different packages of interest. So I think some lessons learned from this was just to like really read the docs carefully and just a lot of trial and error. And then another challenge that I'm still facing is the fact that this code was really inefficient compared to GTEx. I think it took like 10 minutes to run compared to like the seconds you get from GTEx. So that's something to keep working on. Next slide. And then I also learned a lot from this project. I think by exploring this data, I gained a lot better understanding of CFDE tools such as PubMap, GTEx, and Reactome. And then I also gained a better understanding of how to process genome data by exploring these RN Conda packages. So some acknowledgements from this work. I'd like to thank the tissue donors and the UCSD Pregnant Reproductive Tissue Mapping Center for providing me with this data, as well as my collaborators. And I wanted to thank the CFD Training Center for giving me this opportunity.
Thank you, Molly. That was fantastic. If you have any questions for Molly, please put them in the Q&A in the Zoom feature, and she can try to answer those in writing. Thanks again, Molly. So next up, we have Luis Pena Marquez, who could not be here today, but was able to provide a pre-recorded presentation on his study of brain-related traits using predicted brain tissue-specific gene expression in a Mexican-American cohort. Luis is a PhD student and research associate at the University of Texas, Rio Grande Valley, mentored this summer by Harold Goring of UT Rio Grande Valley and Kristen Ardley with the CFD GTEx program. So Bo, please start the video.
Hello everyone, my name is Luis Peña and I'm a PhD student at the University of Texas, Rio Grande Valley, working in the Division of Human Genetics. Today, I will be presenting work from the CFDE Summer Mentoring Program. Thank you for the opportunity. This study was conducted under the guidance of Dr. Harold Goring at UTRGV and Dr. Kristen Ardley from the Broad Institute. Our project focused on using genetically predict gene expressions in brain tissue to study brain-related traits in the South Texas family cohort. The overall objective is to identify genes causally influencing brain traits, such as cognition and Alzheimer's disease. To do so, we have predict genes expression for different brain regions based on the GTEx reference datasets in a South Texas family study. The resulting genetically predict gene expressions value have subsequently been correlated with measured brain traits of interest in this family-based cohort. We have also sought to evaluate the utility of predict-scan predictions for complex disease genes discovery. What is GTEx? The GTEx project is a large-scale resource to study gene expression across many human tissues. Funded by the NIH, GTEx provides a reference dataset of gene expressions profile and EQTLs in diverse tissues, most of which are difficult or impossible to obtain in studies of living individuals. Helps in the characterization and interpretation of the function of millions of genetic variants across the human genome. Helps in understanding the molecular mechanism of genetic risk for complex traits and disease. Reveals tissue-specific regulatory mechanisms critical for interpreting GWAS and revealing disease biology. Enables prediction of gene expression across many human tissues. GTEx recruited 838 in the V8 release version. Recently deceased donors, most with relatively good prior health. The dataset is predominantly European ancestry, with a smaller representation from African American, Asian Americans, and Hispanic donors. This imbalance is vital because predictions model might not generalize equally well to diverse populations. Sex distribution is also skewed, about 2/3 male and 1/3 female. We have an illustration of the 54 tissue types examining, including the 11 distinct brain regions, with sample number from genotype donors in parentheses. In South Texas Family Study, randomly ascertained multi-generational families, more than 2,000 participants. It begins in 1991, and its ongoing follow-up is rich in clinical and neurocognitive and brain imaging data. It has a multi-omic genome, transcriptome, methylation, metabolomics, and proteomics. PredictScan is used to reference datasets to build prediction models. It predicts genetically regulated expression in a target dataset. Also links regulatory variations to traits and disease. Identifies genes likely to be causal for traits. Why to use PredictScan? PredictScan overcomes the lack of expression data in GWAS cohorts, enables genes-based association studies instead of SNPs-based studies. It helps to identify causal gene-driving traits. We have our conceptual study design overview. We have the GTEx donors, and each GTEx donors provide a RNA sequence or transcriptome data plus the DNA sequence or the genomic data. And it combines the transcriptome and genomic data across the 54 tissues and is used to map the tissue-specific eQTLs that explain how SNPs regulate expressions. This forms the training resource for prediction models. So, using the eQTLs, we train the gene expression prediction models. Then, we take the DNA sequence data from the South Texas Family Study and impute it into these models. This allows us to infer expression in tissues that we cannot measure directly in the South Texas Family Study participants. PredictScan uses the genotype data, predict genetically regular expression, and this gives us an imputed transcriptome, right? Tissue from the tissue of interest, without the needing of RNA sequence from each participant. Lastly, we perform a correlation analysis between our imputed gene expressions and our measured traits of interest. Data validation. Here we see a breakdown of the prediction models we use. Each row represents a tissue model across models we see. Between 18,000 and 27,000 SNP per tissue contribute to the predictions. Around 12 to 14,000 genes are included in the models. Roughly 11,000 to 13,000 genes are predicted per tissue. Here, we break down how many SNPs contribute to each gene's predicted expression by tissue. You can see that the majority of genes are predicted with one SNP, and also a significant additional fraction uses two SNPs. Only a minority of genes are predicted with 3 to 5 SNPs. Genes predicted with more than 6 SNPs are extremely rare. This shows that while the models cover thousands of genes, the signal often comes from a small number of strong eQTLs. PredictScan doesn't directly measure the RNA. It imputes expression using genotype and the reference models. A key question for our study is to assess how well the imputed expression matches the actual measured RNA expression. To test this, predictions were compared against measured RNA sequence data from two distinct cell types. We have the peripheral blood mononuclear cells that are easy to collect and are often used in functional studies. Also, we have the neuroprogenitor derived from lymphoblastoid cell lines which are more relevant to brain-related traits, and they were generated in our lab from induced pluripotent stem cells. This comparison gives us a reality check if predict values align with measured ones. Here we have our first example for the comparison. This gene is the CD163 in PBMCs that has a model based on only one SNP, which severely limits predict power. The scatter probe shows that the predict expression values widely vary, clustering into 3 possible levels. The correlation between predicted and measured values is essentially 0. The prediction model is inferior for these genes in this tissue, with barely any variation in predicted expression levels and no correlation with real expression, making it essentially uninformative and unusable. Our next example. Our second example, unlike the previous example, this gene is the ATOH8. It's predicted much better. Even though only 3 SNPs are included in the model, it produces many distinct predicted expression levels, which track the measured values reasonably well. The correlation coefficient is 0.65, including a moderate to strong agreement between predicted gene and actual prediction. Not many prediction SNPs in the prediction model, but there's still potentially 27 different prediction levels. Overall, not a fairly good prediction. Lastly, in the GSTMA gene, with 8 SNPs in the prediction, it captures some of the expression variability, but there is still considerable scatter around the diagonal. A correlation of 0.42 indicates a moderate level of accuracy. While these predictions can be helpful in a large-scale analysis, they need to be interpreted with caution because of the potential for noise. These genes' expression levels are reasonably well imputed. Here, we step back from individual genes and look at all genes modeled. On the left, peripheral blood mononuclear cells. Most genes have moderate to positive correlation, but some are poorly predicted, even negative correlations. On the right, neuroprogenitor cells. A large number of genes modeled distribute is broader, but some predictions reach higher accuracy up to 0.8. The color scale indicate Pearson correlation, blue for negative, red for positive. Not every gene is well predicted. Some genes are essentially unusable, others are highly reliable, most fall in between. It's important because the negative correlations could be random or an artifact, but the fact that there are many more positive correlations, it means that the prediction is working. Here we quantify the heritability of the predicted gene expression traits in PBMCs. The mean heritability is 0.93 and the median is 0.97. This is very high compared to measured expression traits, which often have moderate heritability due to environmental and technical variations. This is what we expect given that the predictions algorithms is entirely genetically driven and explicitly does not consider any environmental factors, which of course do play a role in influencing gene expression levels in reality. Here, I illustrate how we use predict expression in an associate testing. In this example, we want to see if predicted gene expressions in the hippocampus correlates with the hippocampal volume, measured by a structural MRI. The statistical framework. We use a mixed model that partitions variance into polygenic background as genetic similarity across individuals, individual environmental effects. Then we regress hippocampal volume and covariates as age, sex, etc., plus predict expression for each gene. This plot shows the observed versus expected distribution of p-values from our gene-based association test. On the x-axis, we have the expected null distribution. On the y-axis, the observed values. Two curves are showing, blue left hippocampus and orange right hippocampus. What we see? The observed p-value follows the expected null hypothesis distribution. This indicates the following. There is no inflation of p-values. The statistic behaves well, which is good. There is no significant findings, which is not as good. We come to the results. Overview of results. Conduct gene trait association analysis across multiple tissues. We examined cognitive traits and Alzheimer's disease biomarkers. We performed QQ plots to confirm well-calibrated statistics with the strong traits and tissue-specific signals. These slides summarize the complete analysis design. We grouped traits into three main categories: neurocognitive measurement as a cognition score, memory test, processing speed, and facial memory, brain structure measures, hippocampus, amygdala, caudate, cortical thickness, and white matter hyperintensity. Dementia-related biomarkers, tau, amyloid ratio, neurofilament light, and GFAP. For each trait, we ran gene expression predictions in relevant tissues. Notably, we include covariates in all models to adjust for confounding. As age, sex, and education for cognition-related traits, we use intracranial volume and EDIV, which stands for Estimated Total Intracranial Volume for Brain Structural Measures. This ensures that demographics or baseline structural factors do not just drive the signals we found. These tables list the most significant gene traits association across all tested tissues. Each row shows the trait tissue, the gene name, the p-value, numbers of gene tested, the significant threshold, which is the Van Ferroni correction, the sample size, and number of SNPs used to predict expression. For cognition-related traits, we see three notable signals: LSMEM1, ULBP1, and CANXX. All these genes are correlated to Alzheimer's brain and also associated with facial memory. The QQ plots are well aligned with the null expectation, but show sharp deviation at the tail, meaning these are likely true positive rather than inflation. All three traits show well-calibrated distributions with deviation only in the tail. In the gene association with Alzheimer's disease biomarkers, here we focus on the blood biomarkers of Alzheimer's. NT5C2 shows up for amyloid beta ratio. It's linked to metabolic pathways that can influence APP processing. AKAP3 shows up for total tau. And although AKAP3 itself is tested enriched, the AKAP family is directly connected to tau phosphorylation pathways. The QQ plots are otherwise clean, showing no inflation with outliers corresponding to these hits. So the plots show mostly null distribution with single strong outliers. To summarize, we identify novel candidates for cognition, Alzheimer's disease biomarkers, and metabolic traits. Importantly, the QQ plots show clean calibration supporting the reliability of these associations. However, as we will note later, all these results are based on predicted gene expressions, which carries some limitations. One of the biggest limitations is that the most of genes predicted are built on very few SNPs, sometimes just a single one. This reflects the sparseness of EQTL's models in GTEx. For example, in PBNC, about half of the genes are predicted from just one SNP, and in hippocampus, nearly two-thirds are very few. Very few genes have 5 or more SNP in their models, which makes prediction less precise and more sensitive to noise. That's why our significant hits often came from 1 to 3 SNPs only. It doesn't invalidate them, but it doesn't mean replication in larger datasets is essential. These figures show the relationship between GTEx sample size and the number of EQTNs per tissue. There is a clean trend tissue with large sample size like whole blood, skeleton muscle, and adipose yield more than EQTNs, giving stronger predictive models. In contrast, brain tissue like hippocampus, amygdala, and anterior cortex has smaller sample size and fewer EQTNs. This explains why many of our predictions rely only on very few SNPs. Given the limitation we saw, two main solutions are moving forward: expand reference data sets, use differential cells, brain-like cells, reduce study-specific limitations as ethnicity mismatch. Future improvements and directions move beyond the cis-only EQTLs, genome-wide models, use polygenic risk scores, and incorporate trans EQTNs for tissue-specific. Conclusion. Our study demonstrates the feasibility of linking predicted gene expression with traits of interest. Significant hits highlighted biological relevant genes, but sparse niche models limited predictions. Large reference data sets and improvement modeling approaches, genome-wide models are needed. Findings lay the groundwork for more precise tissue-specific gene expression prediction in diverse populations. We have the reference. Thank you so much.
All right. So last but not least, we have Emily Oppold presenting on her mediation modeling of pro-inflammatory cytokines, vaginal microbiome, and preterm birth. Emily is a PhD student in statistics at Rice University, as well as a graduate student trainee at the University of Texas MD Anderson Cancer Center, mentored this summer by Christine Peterson of University of Texas MD Anderson Cancer Center, and Michelle Giglio with the CFD Human Microbiome Project, also known as HMP. So Emily?
Awesome. Thanks for having me. Yeah, as she said, my name's Emily. I'm a third-year PhD student at Rice University, and today I'm going to be talking about mediation modeling of pro-inflammatory cytokines, the vaginal microbiome, and preterm birth. So some of my objectives, sorry, next slide, please. So we'll start by kind of talking about the data source. I used IHMP MOMS PI, going into some project background. Then we'll talk about my activities over the summer, the results, and just overall milestones, challenges, and lessons I learned. Next slide, please. Okay, so to begin, I want to talk about the NIH Integrative Human Microbiome Project, or the IHMP. It represents a large-scale national effort which was aimed at understanding the role of the human microbiome in health and disease. So this initiative spans various life stages and medical conditions to provide an overall comprehensive picture of microbiome dynamics. So they had 3 major studies that were funded by IHMP, and one of those specifically focused on pregnancy and preterm birth, which was called the MOMS PI study. So it's a pregnancy-focused component of the IHMP, and its goal specifically was to longitudinally characterize the vaginal microbiome along with the host, so the mother's, biological responses throughout pregnancy. So this provides a unique opportunity to explore how shifts in microbial communities and immune signaling molecules might contribute to pregnancy complications such as preterm birth. Next slide, please. So, as I said, the main objective of MOMS PI was to understand how both host immune responses and the vaginal microbiome change across the course of pregnancy. And this is important because pregnancy is a dynamic immunological state and fluctuations in the microbial communities can influence outcomes like preterm birth. So the team collected longitudinal samples from over 1,500 women, not just during pregnancy, but also at delivery and postpartum. They also collected paired samples from infants, and overall, the sampling design enables us to ask time-sensitive questions like how early do microbial and immune shifts changes? Are there specific trajectories that differ between term and preterm births, etc. So that leads us to the question that we decided to ask for our work over the summer. Next slide, please. And that question is, how do pro-inflammatory cytokines and the vaginal microbiome jointly influence the risk of preterm birth? And can we use mediation modeling to uncover biological pathways that could link these systems? Next slide, please. So I'll start just by introducing what are cytokines, what is the vaginal microbiome, and what is preterm birth, and why are these important? So to begin, cytokines are small proteins that help cells in the immune system communicate with one another. So during pregnancy, they specifically help relocate the balance between tolerating the fetus and protecting against infection. So when the cytokine levels are dysregulated, it can indicate that inflammation or an immune response has gone wrong, and these complications are linked to preterm birth. So I want to talk about the vaginal microbiome. It's a complex community built of bacteria, fungi, and other microbes that live in the vaginal environment. So I learned over the summer, with the help of my mentors, that in order to have a healthy vaginal microbiome, you need to have a low diversity, but in the sense it also needs to be dominated by the lactobacillus species. So the lactobacillus species helps to maintain a low pH of the vagina and inhibits the growth of harmful pathogens. So disruptions to this ecosystem can be linked with preterm birth and delivery. And preterm birth, I learned, is actually the leading cause of death in newborns worldwide and can carry serious long-term consequences with children's health, development, and economic stability. So despite its significance, it's poorly understood, and so that's why we decided to look into it this summer. Next slide, please. So I want to give a little background into what mediation modeling is. It's a statistical framework for uncovering how and why one variable influences another. So rather than just testing whether an exposure is associated with an outcome, mediation lets us ask the question of whether that effect might be explained by a third variable. And we call this third variable the mediator. So in this context, our exposure, X, is the level of the pro-inflammatory cytokines. These can initiate or signal immune responses. And then the mediator is the composition of the vaginal microbiome, which could be changed based on inflammation. And in turn, our outcome is whether or not the mom gave preterm birth. So an direct effect would just be straight from the pro-inflammatory cytokines to preterm birth. An indirect effect is when it passes through our mediator, which in this case is the microbiome. So next slide, please. But this separation allows us to identify biological pathways and better understand the mechanisms underlying adverse pregnancy outcomes. And it's especially useful in complex systems like pregnancy, where multiple biological layers interact and interventions might be targeted at different points in the pathway. So as you can see here, this is a high-dimensional setting where instead of just one mediator, which would just be the overall vaginal microbiome, we actually went and we chose specific species like the lactobacillus that I talked about before, and it kind of gets a little difficult sometimes with statistical modeling because you can have 10, hundreds, thousands of these different mediators, which all follow a different pathway. So next slide, please. I want to talk about the actual data that we worked with. So we began with 7,665 rows that represented the microbial features, and then we had 29 cytokines. And when we looked at those two samples together and we were able to align them, we had 1,241 matched samples. And so moving forward, when I talk about the data set, I will be talking about these samples that were the same. And when I talk about alignment, what I'm trying to say is we had many subjects, many moms, but these moms came in at multiple times, multiple times throughout pregnancy, and then at delivery, and then at postpartum. So when I say alignment of samples, it's just saying, hey, here are all the times that these moms came in at the same time. So I want to go to our next slide and talk about a microbiome sample summary. And so now we're kind of starting to look at the overall abundance of the microbial features at this point. So our data is highly sparse, with approximately 98.4% of the values being equal to 0. And it leads us to this skewed distribution. So the per feature, what percent of samples have a non-zero value, and the average relative abundance for each feature creates this histogram overall. And so next I'll lead us to the cytokine profile summary. So, sorry, next slide. So we looked at 29 different cytokines that were quantified from biological samples collected over the longitudinal study. And so overall, you can kind of group all of these cytokines into whether they're pro-inflammatory, anti-inflammatory, or if they're just considered regular immune signaling molecules. For our specific research question, we were extremely interested in which one of these cytokines were pro-inflammatory. And so we ran overall a couple different data analysis. And during a literature review, I was able to see like, okay, out of the 29 cytokines we have, IL-1 beta, IL-6, TNF alpha, INF and gamma, and IL-17 were the pro-inflammatory ones. And so overall, after some more analysis, we decided to move forward and look at IL-6 specifically being a pro-inflammatory cytokine for the rest of our project over the summer. So on the next slide, I kind of talk about some key activities. And so we're going to start just by looking specifically at the microbiome and cytokine independently, and then looking at these combination plots and seeing really what the results are, and if these two working together do lead to preterm birth. Next slide, please. So the histogram displays the average relative abundance of each genus across all samples. So each bar here in this diagram represents one genus, and the height of the bar indicates how prevalent that genus is on average in the population. So relative abundance is a proportion, the percent of reads assigned to each genus per sample. So taking the average across samples helps us to identify the genus levels that are consistently abundant versus those that are rare. And as you can see, this is often more of a right skewed distribution. But then we were able to go in and see which ones were abundant. And on the next slide, I actually was able to find the top 10 genus by their mean relative abundance. And you can see that Lactobacillus, which we talked about being the healthy microbial feature in the vagina, was dominant in this type of setting. And then after that, you have Streptococcus and Prevotella, which are not necessarily healthy microbial features. And so we'll talk about how that kind of influences the vaginal pH and the mom's health overall, with the idea of whether or not she's giving preterm birth. So on the next slide, we kind of start looking at the difference between the cytokines and microbial diversity. So most cytokines show weak correlations with Shannon diversity. So we use Spearman's rho, generally below 0.2, to indicate these modest relationships. So the top positively correlated cytokines, which are referenced in the table, include GM-CSF, IL-1 beta, IL-8, IL-6, right? These are the same pro-inflammatory ones that we talked about. And they're all associated with increased microbial diversity. And remember, in this context, microbial diversity being high is not necessarily a good thing. We want low microbial diversity with a high dominance of lactobacillus. So overall, the positive correlations between these cytokines and the alpha diversity indicate that higher inflammatory signaling may be linked to greater microbial diversity in these samples, including IL-6, which is what we used going forward because it is a key pro-inflammatory cytokine and it shows a consistent positive correlation with microbial diversity. So next, I kind of want to start looking at some of the analysis that we did between the two. So this is a PCOA plot. It's based on Bray-Curtis dissimilarities calculated from genus level relative abundances of the microbiome. So each point on this graph represents one sample, and the distance between the points reflects how similar or how different the microbial communities are in terms of their composition. So what's very interesting here is that we were able to find all these groupings, right? All of these dots that are the same color are located in the same region, meaning that these microbial diversities, when they're close to each other, are actually very, very similar. So we were able to see that there is some correlation between these aspects. And on the next slide, you'll be able to see our volcano plot. So what this plot represents, each point here, once again, is a microbial feature. And on our x-axis is our effect size. So positive values means a higher abundance with the variable, and negative values means lower abundance with the metadata variable. So overall, all of these red guys, especially the ones in the top right-hand corner, are high abundance in the exposed group, and they have a strong significance. And then below the significance threshold, so all those dots that are gray, they are not statistically significant, so they were not worth looking at in our study. And then overall, if the center of the plot, which is near x equals 0, these are features that have some effect size. It's just usually very, very small. So they are overall less biologically interesting. So yeah, we'll go ahead and we'll move to the next slide. And I kind of want to start talking more about mediation modeling and why we decided to make our own method. So there are existing methods right now, but they fall short. So most traditional methods were designed for settings with just one or few mediators, where if you remember that slide where I had lactobacillus and streptococcus and all those microbial features, we wanted to be able to create a method that could account for 10, hundreds, or thousands of those. And a lot of popular mediation tools assume that the mediators are independent, which we know is false in biology. Cytokines and microbes interact, and if we ignore that correlation structure, we risk both inflated false positive and missed true signals. And finally, these methods often emphasize p-values alone, and we want to look at correction strategies like local FDR, which is your false discovery rate, and find clear effect size estimates. So that leads us to the novel approach, which is on the next slide. So how does our algorithm help? We cluster the microbiome features biologically into meaningful categories. So first, we have our first arrow, which is the effect of the exposure of the cytokines on microbial features, and then the effect of the microbial features on the preterm birth. So we use a mixture of 9 Gaussian components, where each cluster represents a distinct scenario. So we have mediation clusters, partial effect clusters, and null clusters. So as you can see here, all the ones in the red are the mediation clusters. So both effects are positive or negative. And then in the blue, we have our null clusters where we have at least one arrow is null. So this helps us capture relationships jointly rather than just testing each effect in isolation. And we're able to assign each feature a probability moving forward to adapt to complex real-world patterns. So looking at the next slide, one of the problems that I wanted to talk about and one of my challenges was that the preterm birth outcome is still under controlled access. So we were not able to go in and look and see whether it was true or not if the mother really did give preterm birth. So we had to simulate this outcome ourselves. So we loaded the microbiome and the cytokine data, and then through that, we went with a Maslin 2 procedure, which basically just helped us see where the connections are between these two. We removed low prevalence, no variance, and then we log transformed and scaled this. And then basically, once we had our effects, we chose 10 true mediators, 5 that were positive and 5 that were negative. And we assigned these different effect sizes. And then we actually used this logistic model to assign a probability to these effects. And then we were able to compute test statistics, T statistics for each mediator, compare them to the Maslin 2 results, and create our ground truth table, which basically told us at the end, did this mother give preterm birth or not? So now we're going to go ahead and talk about our results. And then actually, if you'll just go to the next slide, please. Okay, so here we have our picture back to those 9 different clusters. Each point on this graph is put into a different cluster. So you can see the 9 different ones on the side. And I want to be able to tell you guys about our red point here. So this red point came from cluster number 1, which is a positive-positive interaction. And it represents the association between IL-6, which is our cytokine, and Streptococcus, which is our vaginal microbiome. So both of them agree on a positive effect, meaning higher Streptococcus abundance is linked to higher IL-6 levels. So biologically, this makes a lot of sense because the vaginal Streptococcus is associated with inflammation and adverse outcomes like preterm birth. And IL-6 is our key inflammatory marker. So the fact that both the methods agree and our algorithm was able to group this point with similar positive associations show that the model is correctly capturing meaningful cytokine and microbiome biology. On the adverse side, I also wanted to talk about this blue point here, which was a negative-negative cluster. So this point represents the association between IL-6 and lactobacillus, which is our healthy vaginal. So as you can see, that was correctly grouped in the negative side versus the positive side. So overall, on the next slide, the CFDE helped me to be able to write a summary report and publish my first R code on a GitHub repository. And so on the next slide, I'll talk about my milestones achieved. Over the summer, I was able to combine microbiome, cytokine, and metadata from the MOMS PI study. And I really learned how it allows accurate downstream modeling. I was able to do our clustering-based mediation modeling to identify these mediators in these high-dimensional settings. And overall, I was able to implement all of this in R and apply it to a specific CFDE cohort. And basically, on the next slide, I want to talk about challenges and solutions. So one challenge that I kind of already talked about was the restricted access to preterm birth, and therefore we had to go and manually simulate the outcome of whether or not the mom gave preterm birth. The next challenge was integrating the cytokine data with other omics layers. So it posed alignment challenges, especially given differences in sample collection timing and identifiers. So we worked to really accurately make sure that we were looking at time points across data sets and ensuring consistent and reliable integration. And finally, I think one of my biggest challenges was understanding biological significance of cytokine and microbial patterns. I did a lot of like literature review over the summer, and my CFDE mentors were really helpful in the sense of like, at the beginning of the summer, I didn't understand how important and healthy lactobacillus was in the vaginal environment. And now I feel like I can really understand that and other vaginal microbial features. So that leads us to our lessons learned. And one of those is the longitudinal data handling. Being able to look at those different samples over time was incredibly important. And the timing matters, right? Whether or not you were able to take those data points when the mom was still pregnant versus when she gave birth versus postpartum is very important to being able to draw meaningful conclusions. And finally, just as I kind of talked about alignment, I had to develop different strategies to harmonize these data steps. And so that was a good lesson learned for me. And so finally, overall, I was able to use this some skills that I gained on the next slide. Using Maslin 2, identifying the cytokines associated with the key metadata variables. This was a new tool for me. I had never used this before. In addition to, I really was able to embrace my analytical thinking and overall just communication and reproducibility, making sure documenting all the analysis pipelines that I worked through and being able to share it with not just one, but two mentors and others at the CFDE. So that leads me to my last slide, which is just acknowledgments. Thank you to the CFDE for this opportunity over the summer to be able to enhance my professional development. And thank you to my mentor, Dr. Peterson at UTMD Anderson Cancer Center, and Dr. Giglio, who was our CFDE mentor over the summer from the University of Maryland School of Medicine. Thank you.
Thank you so much, Emily, for that wonderful presentation. We do have one question from the audience. You can see it in your panelist chat, but I'll read it for the audience as well. Why did you select cytokines as exposure and the vaginal microbiome as the mediator and not the other way around? Would both orders be possible, potentially even simultaneously?
Do I have time to answer it now?
Well, yeah, let's take a minute and answer it. That'd be
Okay, great. Yeah, we talked about doing it the other way around. Overall, like just the cytokines being like the signaling molecules and all of them always having being able to choose a cytokine that always had the pro-inflammatory characteristic was super important. Because overall, we're hoping that those being pro-inflammatory leads us to an overall inflamed vaginal microbiome, which could then lead to preterm birth. I hope that answers the question.
Thanks, Emily. And if not, we'll ask them to put some follow-up in the chat or in the Q&A for you to answer. Thank you again. And thank you to all of our mentees who presented today. Again, great work over the summer, and we really appreciate you being part of our mentoring program, especially as it was a pilot. So they got to experiment with us and work with us to make the program even better for next summer. So, all right. So next up, we have what we think will be a really interesting discussion about empowering students and researchers to leverage Common Fund data for their professional growth. Our panelists joining us today have all empowered the next generation through a variety of work and are poised to share that with you today. Moderating our panel is Alyssa Dillman, our CFD Training Center co-PI and Training and Engagement Director. And joining us as panelists, we have Jeet Vora from CFD's Glygen program, Sue McClatchy from Jackson Laboratory, and Sue Tappan from CFD Spark. So Alyssa, take it away.
Thank you so much. So yeah, we are going to sort of do a brief overview of some of the training activities that are happening throughout CFDE, and then have a small discussion around sort of some of the things that might come up for training, and we've had a couple of polls and other things that we sort of collected for our discussion session. But if you also have questions, you can feel free to stick them in the chat. But Sue, would you like to, sorry, Sue McClatchy, would you like to kick us off?
Yeah, I'd be happy to. I'm going to turn on my camera with the caveat that my internet is a bit unstable today, so I might have to turn it off. Hi, everybody. I'm Sue McClatchy. I'm a bioinformatics analyst at the Jackson Laboratory. I'm really glad to be here with all of you. Are we going to introduce everybody else, or should I just roll, Alyssa?
Just
what do you
roll.
think?
Yeah.
Just roll.
Jennifer introduced us a little bit, so I think we're okay.
Okay, we're good. So we recently received a grant award from the Common Fund Data Ecosystem to develop a course that we are calling the short course on methods for multiomics data analysis. So I think the big kick is this, that there's a ton of data out there. There is no shortage of data, but that we need to encourage people to reuse data that's already out there instead of generating more and new data. There's a lot out there that has not really been mined to the full extent. So what we'd like to do is to train people to master a diverse set of skills in biology so that they can effectively reuse large-scale multiomics data from the Common Fund. And that's what our course is going to be all about. We're going to be offering that course in April 2026 in Bar Harbor, Maine. And this fall, we're going to gather together some of the Common Fund investigators from some of the data programs that we'll be using to talk to one another and to talk to us and to the trainees that are present about, what skills people need, how best to integrate these data from these different Common Fund programs. And critically, one of the things that we're teaching is team mentoring. So during the course next April, we're going to have a team mentoring professional who's going to guide us through working in teams, which is not an easy thing, right? Because as the cowboy poet Baxter Black used to say, people are funny critters. And working on teams is hard, but science is a team sport, and so we need to learn how to effectively work on teams. We're going to prioritize how to identify, navigate, and access data resources because there's tons of data out there, but it's not really so straightforward getting to those data or understanding what's in them. And then we will be able to train people how to integrate data from those Common Fund programs. And also we'll be putting them in scientific teams. So we're still just sort of getting started and haven't offered anything yet, but we have big plans for later this fall and then again next spring. And I think I'll stop there and turn it over to the next panelist.
All right, thank you, Sue. Jeet, I think you've got some slides you wanted to share.
Yes. Hello, everyone. So my name is Jeet Vora. I'm going to share my slides and then begin with the rest of my introduction. So all right. Can you see my slides now in full screen mode?
Yes.
Yes.
Okay, perfect. So my name is Jeet Vora. I'm the project manager for CFD Glygen project. I'm at the George Washington University in Washington, DC. And today I'm going to talk about the training opportunities we have at our lab, essentially under GW Glygen. And also I'm going to talk about some of the skill gaps in the emerging workforce that we witnessed after having these training opportunities through like a couple of years now. All right, so let me first introduce to you the CFD Glygen internship program. In today's talks, like the early talks in the afternoon, you heard about the resource called as Glygen, which is a glycoscience resource. And what we do is we essentially integrate glycoscience data and connect with the other omics data. It could be related to gene, protein. It could be also related to your transcriptomic factors, so on and so forth. So essentially, we do data integration under the glycobiology domain. So this internship program, we had it in 2024, and then in 2025, it was around for 8 weeks. It was in person at the George Washington University, and it was funded by the CFDE grant. And the good thing about this program was it was a paid internship program, and the students had the opportunity to work on different projects. It could be under data wrangling, bioinformatics programming, developing LLMs to extract data, and some deep learning modules that they can apply in the glycobiology space. And this eventually, the data that we got from these projects was ultimately submitted to the CFD portal through our Glygen data submission pipeline. We have some interesting presentations also after this. The interns, they had the opportunity to present their work to the community, get their feedback. They had opportunity to network with the research scientists, with the subject matter experts in bioinformatics, in machine learning, in glycobiology. So it was a very interesting internship program for the students. The one thing I would like to mention that we had this in 2024 and 2025. It's now discontinued because we don't have the funding from CFDE. But if we get any other funding, we might resume it for the next year and have projects under it. The other thing I would like to point out is we got around 800 applications across US from undergrad and grad students. So there was a lot of interest in the program. So that was about the CFDE Glygen internship. What we have now is the Hive Lab volunteership program. So Hive Lab is my PI's Raja Mazumdar lab. There is a reason why it's called Hive. It's there on the website. So what we're starting with the volunteership program, we started this year in summer, and then we are having it in fall. The fall application is closed now, so we're not advertising it at the moment, but we might have it for next year. So if anybody is interested in doing this volunteership program, I will send the emails that you can contact. So it's a 12 to 13 weeks bioinformatics program. Again, I want to point out we are not a wet lab. We focus on bioinformatics, databases, and tool development. So yeah, that's where we are at. So we provide the remote or hybrid program depending where you are. If you want to do it remote, you can do it remote. If you are here in the area, you can also do that hybrid program. And we have multiple projects that you can choose from, not just CFD Glygen, but you can also work on biomarker KB, which is also CFD funded. You can work on Predict Mod, Argos DB. So these are a few of the projects based on your interest, what you want to do, extraction, LLM development, or AI-ready data sets. You can pick and choose these projects and you can work. So we assign a mentor so that you can work with them, and a point of contact. So anything you want to know, or if the mentor is busy, you can reach out to point of contact and work with them. So it happens asynchronously and synchronously as well because of the mode of how we have developed. You also have the opportunity to present. So this is what we had, or the picture where the volunteers presented, and the previous pictures were from 2024 and 2025. So yeah, this is about our volunteership program. You can reach out to us. And if you do well in the program, yeah, most of the students do. And if we have openings in the lab, we do hire some of the students to work with us, or there is also an opportunity to join the program at GW if you want to continue your studies. So multiple streams down the line that you can pick and choose. So based on our experience, we also had volunteership programs before this, but it was on a small scale. And the volunteership programs also, it was not restricted to graduate and undergraduate students, but we also opened it up for the high school students because they wanted to get into the field, know how the field is about, and if bioinformatics is a good fit for them, if they want to choose a career. So they wanted to just do a trial with us and see how it works for them. And most of them, they are choosing, or they have chosen bioinformatics track in their undergrad or grad studies. So based on the experience of these programs, what we have understood is the students are really great in doing the extraction, the curation, the developing the LLM models, all the technical stuff. They're really great programming. They know how to program. They know how to use the tools that are available. But what we realize is there are certain skills that they need to brush up, or if they want to be part of the workforce, they need to work on those skills, polish them up, and join the workforce, because that is what is going to set them apart from the other candidates or other employees starting with them. So in my next few slides, I'm going to point out to some of the things that we noticed were the skill gaps that we found in the students, and if the students work on them, they can really enter the workforce, without any issues or without any struggles in the workplace environment. So I'm going to present those. And just in the morning, I was reading about a Stanford document or a report which said that the generative AI is going to um, uh, cause issues in hiring of the new employees between the ages of 21 to 25, because most of those jobs are getting done by the AI. So some of these skills will definitely set you apart and make you more hireable. Okay, so the first skill I would like to point out is the verbal communication. So the next generation workforce I'm talking about, they struggle with the face-to-face conversation. They are more into emails, texting. They don't want to do phone calls, or they just don't want to, indulge in small talks, getting to know what the other person is working on, or they're mostly reserved, which can be a problem. So my PI, he always advises me, if there are more than 10 emails, just contact that person, understand what is happening instead of, having those multiple threads. A phone call is always a good thing to keep in touch, and also to get to the bottom of the issue or whatever work you have been assigned. So try to do those. Professional writing. Most of the students that we had on these programs, the writing was good, but sometimes we felt it's not up to the professional level. The professional tone, the etiquette to write, or the business writing skills were lacking. It was more like how you communicate on social media rather than how you need to communicate in a business or a professional environment. Based on my experience, like last year, we had these interns who just joined, and we had a Slack, and we were just trying to explain them the projects, and some of the communications were happening, and they were using a lot of slangs, which of course I was not used to it. So I had a really hard time of trying to understand what they meant. So I had to Google and understand. And I just requested them if we could stick to professional communication. And then they were then following up with that. So just take your own judgment call where and how to be professional. Networking, again, this is a perceived skill gap. Networking is very great to get a job, to get a position, or to even, if you're having an issue, if you know people in that specific domain, you can reach out to them, and you can talk to them and discuss your problem. And many of us and many of the experienced mentors or the employees, they will really go ahead and help you. So yeah, connect with them, and it will be a good skill if you can master it. Emotional intelligence is something where you need to read the room, understand how the workplace dynamics works. So if your PI or your boss is having a stress day, it wouldn't be wise to come up with a good idea, excitingly sharing it with your peer, because that's just not a good day to discuss that. So try to read the social cues, or um, if you're in a meeting, try to be short. If people are getting bored, then try to minimize your communication, or shorten your communication. Conflict resolution, something that if you have your opinions, if you have your ideas, they can be turned down, and you should take it personally. Not you should take it professionally and not personally, because it's nothing personal to turn down your ideas, or if you have your opinions. So yeah. Then comes the adaptability. We work in a very fast-paced environments, and your PI gives you a task today, and tomorrow he might come and say, okay, we're not doing this, we are doing that. So be a little open-minded. You can negotiate with your PI. You've already worked on it. Let me try and see. So, you have to be a little adaptable. And then always take, I would say, initiative and independence rather than waiting for your boss or your PI or your manager to tell you what you need to do. You can just take proactive steps and do certain things. I'm not saying, go all the way overboard with it, but, you can just keep him with the loop of the new things that you're thinking or the steps you have taken. And then comes the professional boundaries, understand the workplace environment, workplace behavior, the dress codes, the communication styles. Some of the people that I worked with, they don't like long emails. So I just, and if I want to catch their attention, I have the words like urgent or priority in the subject, and I just have bullet points and give them the actionable item that they need to do and a due date that they need to get back to me. So something like that. And the other thing, yeah, this is what we experienced some time ago, that we had this symposium, and we had clear instructions about, how you need to prepare for that symposium, how you need to present. And one of the student, she was attending or presenting at the symposium in a park, and there was a lot of noise around it, and it was not very professional to do such kind of presentation. So always respect others, make sure you have your dedicated time for that symposium. You sit in a quiet place and do those presentations or take those important meetings. So I've covered these. Okay, and most of the things I've already covered. You can make, you can go on some apps to, practice your verbal communications. You can network actively. You can develop your emotional intelligence by reading body language guides. You can practice active listening. You can seek mentorship. Uh, Most of you will have some colleagues, senior colleagues who will be willing to provide guidance and feedback. And as I mentioned, take initiative, just ask what else I can help with rather than waiting for somebody to assign you work or tell you what you need to do. You can also ask better questions, or you should be asking questions. Why should I do this? Not just blindly or just take the work that you've been assigned. You have to research about it. What is expected of me to do? Why should I do this? And you should just tell, I think we should do X because why? What do you think? Always pose that question as well. Learn from your colleagues, shadow them. Uh, of course, over the years, they have gained those experience. You can also join your professional development workshops. So we also offer such workshop as part of the internship and volunteership, because again, I said, we believe soft skills development is as important as technical skills. And yeah, do practice some small talk that just helps you understand your colleagues and the workplace environment. So there's nothing harm with it. Again, these are all what we experienced based on the internships and the volunteership programs that we had. And this is what we felt the interns or the emerging workforce, which are the students, they should practice and follow. So yeah, that's all I had.
Thank you so much, Jeet. All right, can we have Sarah, sorry, Sue, ooh, not Sarah, Sue, Sue Tapan next from Spark. Oh, you're muted.
There we go. Okay, I think I'm all set now. You can hear me?
Yep, thank you.
Awesome. Okay, so I'm gonna just give you a brief intro on how the Spark Data and Resource Center thinks about the amazing research that is being done in the biomedical community and the unique ecosystem that we're building, and that we're hoping that everybody who's affiliated with the CFDE and beyond begins to dive in and play with. So our goal is to really make this data that we provide accessible, operable, and reproducible. And the way that we do that is that we've got it's a trusted cloud-based platform for sharing multimodal data, and we meet the NIH, HEAL, and journal standards for scientific data repositories. And so all of the data that is contributed to Spark adheres to a uniform data set structure, whether it is computational code that is provided, microscopy image data, micro CT, physiology, et cetera. They all have a similar foundation of organizing structure to allow people who are not the data contributors an insight into how the data is created, how it is organized, and the formats in which you can use them. I know that when you start using data that you didn't create, part of that learning curve is just getting familiar with what variables were collected, what is the common data elements that are being utilized. And so the 350 data sets, models, and simulations that are present on the Spark portal all have all of that provided as part of the uniformity that we enforce across the Spark portal. So if you go to the Spark portal, which is spark.science, you can see that we've got a number of different options up at the top. And one of the things that I want to just touch briefly on really quickly are 3 Spark apps that are available for everybody to use and may have relevance for you as you're building your own research program and getting your feet ready for your own experimental domain. And so Spark has a number of ways to interact with the data and models that are present on the portal. So what I'm showing you here is our anatomical flat maps. And so what they do is they incorporate SCAN knowledge. So SCAN is a sophisticated knowledge base that integrates the neuronal connectivity of different neuronal populations that underlie peripheral organ control. And so they're automatically drawn on the flat map. And you can see here that you've got all these little individual number markers that are popping up. Those indicate that there are data sets or models that are present related to those particular subjects or topics. And so if you were to select a single area or a single pathway, you can see a lot more information about that because it is incorporating not just the individual representation of a kidney data set, for example, but we also have underlying it the relevant phenotypic information about the neuronal populations that help control the kidney. And so in this case, what you can see here is that depending on where you hover, so if you hover over the right kidney, you could see how the populations in the DRG traverse through various nerves in order to innervate the kidney. But if you look at the left, you can see that there are different neuronal populations. So there's an asymmetry in how that neuronal connectivity is actually conferred to various organs. This is really important if you're interested in neuromodulation or just wanting to figure out what types of information you're likely to encounter. And so as we look deeper into the opportunities for developing neuromodulatory devices and other control mechanisms, it's very important to understand the nerves that underpin that information. And so SCAN is also being built upon to create this natural language interface. And so the natural language interface is a way for us to be able to utilize an LLM to query the knowledge base and pull out the information that relates to the kidney, for example. And so you can see how it's drawn and you can see exactly how the information is actually provided within SCAN. Another very important and helpful part of the Spark ecosystem is OSpark. It's a cloud computing platform that is very powerful, and it's available for you to use free of charge. You can request your own account just by dropping an email to support@ospark.io. And you can utilize a lot of services that have already been created, or you can create your own. So we have over 85 created already that cover everything from a Jupyter Lab notebook that encapsulates Neuron to cardiac control models. There's also segmentation algorithms, there's TensorFlow, there's all sorts of different prepackaged building blocks there that you can use to then apply to your own data to establish different and test different scenarios. As a part of the ways that we introduce and try to bring people into the awareness of the type of data models, services, and tools that we offer, we also provide tutorials. So there's a number of step-by-step tutorials that are available on our YouTube channel. They're available in written documentation as well. And in this case, they were featured in a Spark seminar. So you can understand sort of the use case for the tool, why it was created and its real world applications. You can see the tutorial itself and then run it for yourself on OSpark. And that is precisely what the Spark Codathon teams have done throughout the years. So the Spark Codathon has been has invited teams from all over the world to come together and explore and build something new. So we've done this for 5 consecutive years. It's been such great fun. And we have a lot of unique data. We've got a lot of documented access APIs and those powerful processing tools. And so we invite the community to come in to create tools that address their specific research needs. I heard in previous presentations that some of the challenges that can access or be a challenge when starting to use other people's data, so openly accessible data, is that there's various formats, and unifying things can be a big hiccup and a time sink. And that was actually one of the targets of a winning Codathon project this year. So one team created Spark Fuse. They created a data format that would take data sets that were available on Spark, unify convert them into a standard format, so then you could run your pipelines on. In addition to those types of codathons, which are all available on our Codathon GitHub page, they're listed as tools and resources on the Spark portal themselves. So you can explore the Spark portal, check it out, and see each project, watch their video, access their tutorials, and attribute them as well in any way that you would like. But we also provide mechanisms to sort of invite collaboration. So you can imagine that neuromodulation is going to bring together people who do computational simulations, to biophysical modeling, to experimentalists, to device creators, and hardware components all into the same location. And so we have weekly office hours to help people get familiarized with the data that's available. We also do this Bridging the Body and the Brain seminar series. We're recruiting new speakers. So if you know anybody who would like to participate, please drop a line. But we also are continually providing tutorials, use cases, and we'd like to hear from people on what they find useful or could use a little bit more shine up. And I invite you to join the newsletter if you'd like to have an opportunity to sort of stay in touch. But it's a very brief run through of some of the options that we have, so you can get your hands right on the data. So I hope that was helpful.
Awesome. Thank you so much. So thank you to both of our Sues and Jeet. And I'd like to ask if the audience has any questions for any of our folks who've talked about sort of the training and other community engagement activities that sort of, this is just a snapshot of what's happening throughout some of the CFDE programs. Any questions from the audience? Otherwise, I will throw out one. All right, I don't see anything so far. So I think I might ask one question. So the next big thing that is sort of jumping up in all sorts of places and in all sorts of realms in our normal lives is AI. But I think a lot of folks are very interested in sort of how to learn more about how to utilize these tools for bioinformatics or health research. And I'd be sort of curious to hear, how researchers can at least kind of scratch the surface or learn a little bit more about how to integrate those things in ways that are safe and effective. Oh, we do have a question. Hold on, sorry. I would definitely prefer questions from the audience first. All right, sorry. We do have a question for Sue Tapan. What are the data usage guidelines on Spark?
Yeah, that's a great question. All Spark data models and resources are available under a CC by 4 license, and so you can access them. We expect and hope that you will also attribute, give proper credit to the data and code that you use by either forking the code and acknowledging it, including it in your citations. Each data set includes a tab called, I think it's called citation or reference, where you can just copy the reference themselves. So you can just drop it right into your paper or a presentation to indicate where you found the data.
Awesome, thank you, Sue. And Jeet, there's a question for you. Can a postdoc trainee attend the workshops?
Yeah, certainly they can attend it. It's open to everyone from high school students to postdoc. Anyone can join the workshops, even the volunteership program if they want to use data from a particular project, or they want to advance the particular project. Yeah, they can definitely join. Yeah, we will have some details. We regularly post the workshops and the other activities on our portal. So, they can check on the portal, or they can just I can put my email address and they can communicate or contact me.
Awesome, thank you. All right, back to our AI question.
So I would like to address that. I mean, the question is about using AI and using it effectively and responsibly. And so I use it all the time. But there are situations when it just really isn't appropriate, and that is when you're trying to learn something. So I recently saw a comic from a comic artist who just so happens to be a Columbia University professor, who developed this comic to present in a class that he's teaching this term, to explain to his students why generative AI is not a good idea for them to use in learning. And if I can quote it, so it does not let you experience the struggle or the joy that you need to have in order to learn something. It robs you of experience, and it conflates answers with learning. It will give you all of the answers. Your brain did not have to do any of that work. So because you didn't have to do any of that work, you really don't learn. Anyway, I don't know if that said, there are great uses for it. There are great uses for it in completing code, in checking code and testing code. If you don't know how to code, you shouldn't really, you don't want to start by having generative AI do it for you, because you miss out on the struggle, you miss out on the joy. You need to experience all of that in order to learn. Just having the answers given to you is really not helpful to you to grow personally or professionally. I'm like right now, I'm going to tell you, putting my money where my mouth is. I'm learning about metabolomics right now. I don't really know. I'm not an analytical chemist. I really don't know much about analytical chemistry. And yet I'm making myself slog through papers, books. I am doing the hard work, and it is mind-boggling. It really is. But I will not turn to a generative AI to have it write for me. I need to write about my metabolomics from a place of knowledge and not let it do it for me. I'm doing the hard work, and it's you know, in some ways it's kind of exhilarating. It's hard, but it's exhilarating. So that's my 2 cents regarding AI.
I'd like to pop in and just share, I agree that you have to know the limits of what generative AI can really operate within. And as we were creating Scan NLI, it does use a large language model, but it is pointed to our knowledge base. So our source of truth, which it comes from a human in the loop, painstaking, reference, like you were saying, going to those papers and understanding it. And so, if you ask the same question of ChatGPT 4, what are the nerves that go to the ovary? And you ask that same question in Scan NLI, you'll get wildly different things, because generative AI can and will combine sources in ways that don't make sense, right? So when and there are animal differences, for example, species differences between humans and rodents, for example, on what those pathways are. And so having that awareness of the information that you are looking to utilize it for is a big big starting point, I think. One of the Codathon projects, though, actually made synthetic data for AI training. So they took data sets that were available on Spark and then also utilized those to create new synthetic training data for making the model more representative. And so I think that there are so many different ways that you can utilize AI. It is very powerful, and we look forward to using it in more and more places. But the generative aspect of coming up with the question and looking to know whether or not the answer that you get back is a good one is a learning curve for all of us.
Yeah, and if I can add something to it, our lab has actually embraced the use of AI. Well, not just ChatGPT, our PI has given the freedom to use ChatGPT, but most importantly, in terms of research, we are using the retrieval-based AI for developing LLMs and RAG models and deep learning modules. So those tools are really powerful to expand your research. You cannot curate, like, say, 10,000 papers of the field and try to extract information. LLMs are really good in doing that. If you have the structured query, they can do your job really easy, and you can get those missing information from the papers now into your database. So that's just one use case. The other use case is to identify the structures. For example, you heard about it in the next set of talks, how you can identify glycan structures from the papers using deep learning modules. If you go and do that like manually, it's going to take you many years, and there could be a possibility of making a mistake. So it's just helping you to expand your research, and then just ensuring that mistakes are not committed. But yeah, I also agree with Suzanne that you shouldn't be totally relying on generative AI. You should use the organ, the brain, to ask questions, to research for the answers, and not just get the pre-made answer that ChatGPT gives it to you.
I think it's worth pointing out that both of you mentioned basically this idea of human in the loop, right? But in order to be the human in the loop, you need to have a pretty decent understanding of what's happening in the loop. So yeah, thank you for that discussion. And I think that we are sort of past our time already, but the panel will be here for a little while longer. If you have other training questions or programmatic questions, or you're interested in learning more about how you can learn more about these programs, please feel free to reach out to us at any time. Thank you so much.
Thank you all. We appreciate the time you took for that discussion and also sharing all of your experiences and resources from the summer internships that you've done and the codathons and all of the resources available. So next we are going to move into our lightning talk presentations. The lightning talks are from CFD interns and fellows that participated this past summer across some of the CFD internships, including Glygen. So shout out to Glygen for several of our presenters being from that program. The presenters were challenged, which as we've seen all day, we've all been challenged to stay in time. So we've especially challenged our lightning talk presenters to work within a 5-minute time frame and up to 3 slides. So our first person up here is Shubham Agarwal. Shubham is a senior undergraduate computer science and engineering student at IIT Godna. I'm going to say this wrong and I apologize. Godnagahar, India. As a 2025 summer undergraduate research fellow at Caltech, he worked on developing an agentic LLM-based framework for extracting structured biomarker metadata from biomedical publications in collaboration with the EDRN CFD team. So Shubham, you are up.
Yes, thank you very much. Am I audible to everyone? This summer I had an opportunity to work at Caltech with Dr. Ashish Mabal, Sean Kelly from JPL, and the EDR and the biomarker KB teams. In our project, the LLM-driven intelligent biomarker data extraction, we explore how large language models can help transform verbose biomarker descriptions into structured and usable knowledge base. Next slide, please. So the NIH's Early Detection Research Network, or as we know, EDRN, it maintains a large biomarker database hosted at NASA, JPL, and Caltech. Now, this database is incredibly valuable. It contains over 1,000 cancer biomarkers, but much of its metadata is verbose, and it is written as long free text descriptions, which are extracted directly from scientific publications. This richness is very useful if you're an expert studying a biomarker in detail, when you want all the nuances and supporting context. But when you need just specific information, for example, the condition a biomarker is associated with, or a biological role, or the specimen in which the biomarker is measured in, especially for automated systems, this is important because they need to process this information at scale. The verbosity becomes a bottleneck. So right now, these fields are buried inside verbose prose, and manual curation to extract them is both slow and limited in scale. So that's the gap we are addressing here. Next slide, please. So to achieve this, we designed an LLM-based pipeline where the input is a verbose biomarker description and the output is a structured set of fields. And now the main part of this project was prompt engineering. We realized very quickly that just providing a model with a biomarker description and asking the fields, asking it to generate the fields for us wasn't just enough. The outputs were very inconsistent, sometimes vague, and most importantly, they hallucinated details, which is very critical in our case because we cannot let that happen. So we invested our effort into carefully designing prompts, and at the end, this turned out to be a very huge difference. Specifically, we had to define the model's role clearly and tell what it needs to include in its responses and what not. We had to define strict output formatting rules when we wanted how a certain output should look like. And at the end, we also had to provide a few short examples to tell the model how an ideal output should like for a particular input. Now, in practice, without prompt engineering, the outputs were scattered and unreliable. But with it, we achieved consistent structured results that aligned with the schema required by the biomarker KB's team. Next slide, please. Yeah, no. So here we can see a concrete example. So this is a biomarker example, the DCP, the biomarker ID number 19 from the EDRN database. And we can see that it has a very verbose description mentioning its precursor role and presence in the hepatocellular carcinoma patients. So now with our approach, the model was able to extract the structured fields, the condition as the hepatocellular carcinoma, the status as elevated serum levels, the role as diagnostic, and the specimen as serum. Now this demonstrates how a verbose text passage can be distilled into concise schema-ready metadata using with the help of the LLMs. So talking a little bit more about the schema. So we had each biomarker entry given as the input to 4 separate LLMs, which were prompt tuned and fine tuned to to give the certain outputs related to the condition field, the status field, the role field, and the specimen field. Next slide, please. Uh, yeah, so in summary, this work shows that large language models, when guided carefully with prompt engineering, they can enrich the biomedical databases that are already existing out by extracting structured knowledge from the verbose metadata. Now, so far, in the EDRN database, we have only focused on public and fully curated biomarkers with EDRN, but the database they have in the EDRN, they contains many more biomarkers in different stages of curation. Now, looking forward, a key challenge will be to scaling this approach to the full set, eventually extending it to even work with directly scientific publications. And as we do, keeping the process hallucination free will always remain a crucial goal because in biomedical research, accuracy is everything. Thank you very much.
All right, so we'll move to our next presenter. Okay. Oh, yeah, there we go. So Manjul Pradhan is a PhD student in computer science. Manjul is presenting today on estimating the hidden proportion of ligand banding. They're a student in computer science at the University of New Mexico. This summer, working as a research intern in the Translation Informatics Division under the guidance and mentorship of Dr. Jeremy Yang and Dr. Praveen Kumar on the projects in the field of computational biology and drug discovery. So Manjul, when you're ready.
My name is Manjul Pradhan, and I'm a PhD student at the University of New Mexico Translation Informatics Division. Today, I'll be presenting my research on estimating the hidden proportion of ligand binding residues in unlabeled protein sites via positive unlabeled learning. So identifying the binding sites and residues on a protein is an absolutely crucial step in designing drugs and understanding the protein functions. So that there has been a traditional methods which are considered a golden standard, such as X-ray crystallography. But the problem with these methods are they are time consuming as well as they are expensive. And scientists have come up with a different computational methods that are that have shown a remarkable performance in detecting these binding sites. However, the main problem with these methods are they make a common but a flawed assumption that all the residues that hasn't been annotated by a data set are non-binding residues. So in this study, I want to apply positive and unlabeled learning to detect the hidden proportion of binding residues among the unlabeled data sets. So my goal is to tackle this problem with a machine learning approach. So we want to classify these unlabeled residues without simply assuming that all the non-binding sites, all the unannotated residues are non-binding sites. So I'll be using the PulseNAR and the PulseCar algorithm developed here in the informatics division at the University of New Mexico. So how did we do this? So for this, I took a BioLib data set. So for annotating each of the residues, I took all the residues that was annotated by BioLib as a positive sites and all those other that hasn't been annotated as unlabeled data set. Then I applied the PulseCar and the PulseNAR algorithm to validate the study. I had to come up with my own way of evaluating the outcome, the result of these algorithms. For that, I took some of the positive residues from the BioLib dataset and flipped their label to unlabeled and hid them among the unlabeled residues. And so with this, I was able to detect most of the hidden positive residues among the unlabeled data set. The next slide, please. Oh, yeah. And the other method to validate the study was I took the positives identified by PulseCar and PulseNAR to see if they help to improve the performance of the XGBoost. Next slide, please. So in this slide, I'll discuss about my results. So as you can see, in the alpha estimation, both the PulseNAR and PulseCar algorithm were able to estimate the alpha value that was pretty close to the ground truth. Even on the data set that contain only those residues that binds to RNA ligands, the alpha value estimated was almost equal to the true alpha value. And regarding the performance of the XGBoost, both PulseNAR and PulseCar help to increase the performance of the XGBoost, especially with the PulseCar. The accuracy increased from 71% to 91%. So this shows that both PulseCar and PulseNAR algorithm, both the positives identified by both PulseNAR and PulseCar can be used to improve the performance of other computational methods. So in my future work, I want to look at the positives identified by these algorithms and see if they belong to the same protein chains. And also, I want to use the embedded features in the future. That's all from me. Thank you.
I'm good.
Okay, next up we have Campbell Ross, who will be presenting on deep learning for link prediction in glycan images. Campbell earned his MS in the bioinformatics program at Georgetown University. This summer, he worked on his research project as part of the CFD Glygen internship. So Campbell, when you are ready.
Okay, thank you.
Uh-oh.
Yeah.
I was going to say I don't
Yeah.
think those are your slides. So
Yeah.
just a minute.
Okay, so as mentioned, I interned with Glygen this summer, and one of the main tasks of Glygen is to bring more of these glycan structures into databases so that future research can associate them with succession numbers and build up information related to these structures that we find in manuscripts. So I interned under the Edwards lab, which already had done great work in building this glycan image extractor tool. So what it does is it goes through a manuscript that the user uploads, and it finds all the glycan structures in it, So these structures have a standard nomenclature where each monosaccharide in these branching carbohydrate structures is represented by a shape and a color. And then sometimes in these images, there are also linkages, which is two figures, the alpha or beta, and then a number. So that's the anomeric configuration and the carbon bond position connecting those two monosaccharides. So in the current version of this extractor tool, the linkages were not being extracted, only the monosaccharide types. So because the linkages affect the structure of the glycan, that in turn affects the function. So if that information is in the image, we would like to be able to extract that. So my task this summer was to improve this tool to extract the information. And then if it extracts it correctly, we would see that those linkages would actually appear in the extracted version of the glycan, and we would know that that information is getting into the database correctly when we curate it. Okay, so next slide, please. So how did we do this? So first we needed to generate data, and we were not taking images from manuscripts. We were synthetically generating our data by randomly sampling from GlyToucan. All the accessions in GlyToucan, as long as they were composed of common monosaccharide types. And the advantage of this is that we also knew the exact location, like coordinates, and known monosaccharide and linkage types for all those objects in the image. So we were able to use the coordinates and labels of those known linkages in the images to serve as our ground truth when we trained models to predict these linkages in the images. We were also able to display images with and without the linkages shown. So we had some flexibility in making the model robust to both of those kinds of images. The specific model we were using to learn to predict these linkages was the YOLO model, which is a convolutional neural network, originally trained to identify objects and images like general types of objects like dogs, bicycles, and cats. So what we did is we took that pre-trained model and we introduced it to our data set of glycan images, so to fine-tune it to specifically recognize these linkages in these glycan specific images. So for example, figuring out that those are alpha-6 and alpha-4 linkages and reporting those back correctly. And we found good performance in our model, but we were able to do a few things to improve the model and optimize performance. So first, we found that if we added images without the linkages shown, then the model's performance on those specific types of cases would improve. So if you look in the bottom left graph, the orange curve in that precision recall plot did not have any of those images without linkages in it. And when evaluating on those specific types of images, we could see that compared to the blue curve that had 10% of images like that in the training set, the performance was inferior. So adding just 10% of images without linkage info helped the performance improve on those specific images, so we can handle those better. The next thing we found was that adding more images into the training set helped the model generalize better and improve performance on images outside of the training set. So you can see different models in this precision recall plot in the middle graph. Each color represents a model trained on different number of images. You can see that the orange curve with just 5,000 images was lagging behind the performance of the other models. And once you got above 10,000 images, training seemed to plateau, suggesting that for better generalization, at least including 10,000 images in the training set for this specific type of task, achieved optimal results. I will also note that both of these precision recall plots are zoomed in, so the axis starts at 95%. So all of these models are performing very well to begin with. Okay. And then lastly, if you look at the plot on the right, we found that because of there's some linkage types that are very rare in the GlyToucan data set and also in biology in general, but we still want to get those correct when they come up, because if just one linkage is wrong in the structure, if we get it wrong, that whole structure is, when it goes into the database, would be wrong. So we want to get every single link correct in the structure. So we found that we were able to we generated random images with random linkages for every link in that glycan, which means that the very rare types would be more represented because they would come up an equal number of times. So by adding more of those images, and you can see that from going left to right in that figure, in that you're looking at the F1 score on the y-axis, and each of those models has more of those randomly generated linkages, meaning it has a better representation of the rare classes. And we found that getting the rare classes up to about 2% of the class representation, so overall linkage type, helped get that performance. You see the performance improving with each increase in the amount of rare linkages represented there. So those were three things we found to help improve these model performance. And if you go to the last slide, please, we can now see the end result of all this is with that model introduced into our pipeline, the image extractor tool can now extract the linkages in these glycan structures in these manuscripts. So you can see that the original image is there on the top, and now the extracted glycan by the extractor contains those linkages in the image. So that will help improve the amount of information that we're able to get into the databases and expedite that biocuration process. Okay, thank you.
Okay, so next up, we have Cirrus Chun Hong Ao Young presenting on mining glycan biomarkers from publications with AI. So very timely considering our panel discussion. Cyrus is a bioinformatics researcher with a foundation in molecular biochemistry and chemistry. Recently completed his MS in bioinformatics and molecular biochemistry at George Washington University. This summer, he worked on his research as part of the CFD Glygen summer internship. So Cyrus, when you are ready.
Thanks, Jennifer. I'm Cyrus Ao Young, CFDE summer intern under the supervision of Professor Rajan Mazumdar, and I'm sharing my project on mining glycan biomarkers from publications using AI. And just a brief primer, glycans are chains of sugars decorated on proteins and lipids, but they are not just simple decorations, as they can regulate immunity, cell signaling, and even how pathogens invade our bodies. And the fact that they often change in diseases make them useful biomarkers for diagnosis and prognosis. But here's the challenge. Knowledge of glycans is scattered across a sea of biomedical literatures, mostly represented in unstructured text. That makes them very difficult to systematically search or connect the dots. And also, given the fact that the glycans are represented in different nomenclatures and common names, make them hard to standardize and be mapped to an accession. And this project exactly aims to address this gap. So I've developed this pipeline that leverages AI and large language models to extract glycan disease relationships from the literature. So each of the extracted relationship becomes a record designed to be integrated into knowledge graphs containing rich annotations such as which glycan, which disease, along with other metadata including proteins and tissues, and also linked PubMed references. This means that the results are not only properly indexed, but also evidence-grounded and machine-readable. Next page, please. So the core of the glycan biomarker extraction workflow is the large language model extractor engine, which forms the second step of this workflow. So the extractor utilizes the robust OpenAI GPT 4.1 model. This module identifies any mentions of glycans, diseases, and their relationships in the biomedical text. It features a machine-readable output. For instance, if a paper states that the fucosylated N-glycans is elevated in breast cancer, the extractor will group both the glycan and the disease entities and determine their directional association, whether it's an increased level or decreased level. And afterwards, there is this ontology mapper that standardizes and maps entities to ontology databases, including our glycan structure dictionary, and also currently the disease ontology. This is where the AI's ability to make decisions is also being featured, where the LLMs are connected to tools that can query public ontology databases. So in this example, the LLM serves as like the brain of this ontology mapper, receiving a list of entities such as diseases, and it will decide which ontology to query using the two nodes on the right-hand side. So the tool will then return a rank match to the query, where the brain will then analyze the results and decide which is the best match to the original entities. So the biomarker dataset will then be deposited at biomarker KB after post-processing, which biomarker KB is another CFDE sponsored project that incorporates public biomarker data into a knowledge base. Next page, please. So the results on the left shows the top glycan terms extracted from this pipeline, and the pie charts to the right shows a variety of other entities, including the disease that is collected. So, and there's a total of 4.5K abstracts that are being processed, giving more than 4,000 well-annotated glycan biomarkers. Those are informative glycan structures that shows directional changes in observed levels in a cell or tissue sample associated with a disease or a condition. And at a higher level, this work demonstrates how literature-mined glycans can be turned into what's called AI-ready datasets. And by creating a network of biological and disease entities that are standardized and are mapped to ontology accessions, we are enabling AI to traverse and extract knowledge across documents using a technique called RAG, something that both George and Sue already touched on earlier, like specifically graph RAG for discovering glycan disease associations that are linked indirectly and conceptually. So hopefully in the future, this generated work can contribute to the CFDE by enriching first the glycan's annotations regarding the glycan's cellular localization and disease associations, while another work could be done to link glycan biomarkers to the glycan biosynthetic pathways through integrating glycoenzyme expression data from maybe GTEx or IDG, which are part of the other 14 knowledge centers that connects to genes. So I would definitely also try to implement data center APIs in the upcoming analysis, because sometimes the extraction data can be large. So I'd like to close off by thanking the CFDE for providing this invaluable opportunity. This internship allows me to explore the data that CFDE offers, and I've learned how a data ecosystem can spark research collaborations and facilitate scientific discoveries. Thank you all.
Thank you, Cyrus, and we do have a couple of minutes for questions. Oh, Alyssa, do you have a question?
Yes, I do. So sorry if I missed it, but on your graphs here, your pie charts, sorry, quite a bit of each of them has sort of these other categories, and I'm sorry if I missed like what sorts of things are hanging out there in other.
Right, yeah, that is exactly one of the reasons why I was trying to map them to ontologies. So there is a really huge variety for each of the terms. Like for the disease term, it can be mapped to the disease ontology, but the mapping of the other fields, including the glycoenzyme and the specimen type, is still under progress. So the gray areas represent all the other terms that are like not mapping to there's not a lot of terms that are mapping to the same accession. So that constitutes the large proportions in those pie charts.
Okay, gotcha. Sorry, I kind of missed that. And is sort of the eventual goal here, since you're sort of like trying to connect all of these different pieces of information to build sort of like larger knowledge graphs?
Yep, that is right. The current work is to properly map all those fields, all the metadata to their corresponding ontologies, so that it can be better integrated into other data sets using controlled vocabulary. And for example, for the specimen type, we can also use the Uberon, and for organisms, we can use the NCBI taxonomy IDs for etc. So that is the work that is currently going on.
Awesome, thank you.
Great. Thank you, Cyrus. Thank you, Alyssa, for that question. So last but not least in our lightning talks, we have Yushin Zhu, who is presenting on biocurating machine-readable glycan data sets. Yushin is a rising senior at Cornell University studying human biology, health, and society. This past summer, she participated in the CFD Glygen internship, where she deepened her knowledge in glycobiology and developed an appreciation for the role of structured data in uncovering novel biomedical insights. So Yushin, when you're ready.
Hi everyone, and thank you for the introduction. My name is Yusin, and I'll be presenting on behalf of my colleague Anna and I on biocuration and data standardization to generate machine-readable glycan datasets. And this was under the guidance of faculty from George Washington University of Georgia. And next. Yeah, so building on what my colleague has already shared, glycobiology again is the study of glycans, which are sugar molecules attached to proteins and lipids. These glycans play key roles in cell communication, immunity, and disease progression. In my section, I'll focus on glycans, glycosylation patterns, and data resources to highlight both the biology and also current gaps in how we study them. On this slide, we have an overview of many different ways that glycans can be represented in biology. So starting in the top left, we see proteoglycans, which are proteins that create long glycan chains. And this is important as part of the extracellular matrix and signaling. For cell surfaces, proteins can also be modified with glycans, known as glycoproteins. They can either be N or O glycosylated, which affects the protein stability, folding, and recognition. And in the bottom of this image, we see O-link glycosylation, which means that the sugar attaches to the oxygen atom of amino acids serine or threonine, and N refers to the attachment to nitrogen of asparagine. Most importantly, alterations can also be associated with many different diseases, but the main takeaway here is that glycans are not just decorative sugars, they're deeply integrated into almost every aspect of biology, from the host-pathogen interactions to cell signaling and also metabolism. However, despite their importance, glycans are often understudied compared to genes and proteins, and this is largely due to the complexity and also the lack of standardization, and creates a barrier for researchers who wants to use glycan data computationally. Currently, the notation of SNFG are used. You can see in the legend below in the image where each shape and color represent a different monosaccharide. And for example, the green circle is mannose, and these standardized symbols makes data sharing a lot easier. On the right, there is this glycan structure, which has been registered with the glytugan accession ID. Having a unique identifier like this allows researchers to reference the same glycan, regardless of the study or literature that it came from. And beyond identification, databases like Glygen also plays a central role in storing and integrating this glycoprotein and glycan data. Next, please. Yeah, so from this paper alone, we extracted 223 glycans in total, and of those 155 were completely new and require glytugan IDs, which means that 30% of these glycans are currently represented in Glygen, and it highlights this coverage gap and the importance of biocuration. Our project aims to help close this gap by curating and standardizing glycan data into formats that can be shared and used across the scientific community. Next, please. So our workflow starts with carefully selecting relevant papers and high-impact papers in glycobiology. From each paper, glycans, glycoproteins, and compositions can often be presented in images and figures, which are not accessible. After grouping glycans by related figures, they're extracted using tools, including Grid's Toolbox, GNOME, and also uploading by composition. Next. And this is an example of a tool, Grid's Toolbox, that we use to extract those glycans. If the paper are presented with glycans alone, we group them into collections, which essentially are groupings based on shared characteristics and functional features. If the glycans are linked to proteins, we retrieve the corresponding UniProt ID and map the glycans to specific sites and group them accordingly. After extractions and grouping, each data set is revised, standardized, and also uploaded for public repositories and databases such as Glygen and CFDE. Next. So here we see a representation of a dataset with glycan structures with accession IDs assigned, and which makes them accessible and machine readable. Next. And across 9 publications, we were able to curate 1,074 glycans and 84 glycoproteins. Of these, 829 were unique glycans. Of that, 368 were newly registered with glytugan IDs and 461 already existed. This effort expands the glycan coverage and strengthens integration with resources like Glygen and the Common Fund data ecosystem. Moving ahead, we want to move in several directions. First, we hope to improve data standardization by adopting universally accepted formats like SNFG and expand the glycan data coverage by curating papers with focuses across organ systems, diseases, species, and many more. We want to promote accessibility and usability by ensuring that curated data are machine readable and publicly available. And from the curated data, we want to incorporate them into therapeutics to enhance clinical relevance. For instance, using the data in understanding disease-associated glycan patterns and also biomarker discovery. And ultimately, we hope to lay the groundwork for the automation and training for larger scale extraction and analyses. And that is the end of my presentation. Thank you.
Thank you so much, Yusin. We do have 1 minute for questions. I did have a quick question. The resources that you mentioned using GNOME and the toolbox, are those within the Glygen program? Is that how you access those?
Yes, they were developed by Glygen, and their main purposes is for the extraction of glycans. And so there's different, each one serves a different purpose and method, but their main purpose is to extract the glycans.
Okay, thank you very much.
Yeah.
All right, seeing no other questions, thank you so much, Yusin, and thank you to all of our Lightning Talk presenters. You all win today for staying on time and keeping within your 5 minutes, so thank you for that. Again, we really appreciate your all's presentations, and this was fantastic. I am excited to close out this event with our Training Centers Program Officer, Sahana Kuke. She is a program leader in the Catalytic Data Resources team at the NIH Office of Strategic Coordination within the Common Fund. She leads initiatives in multiple Common Fund programs, including precision medicine with AI, integrating imaging with the multimodal data, replication to enhance research impact initiative or the replication initiative, the Common Fund data ecosystem, and the stimulating peripheral activity to relieve conditions, which is also the what we've been referring to as SPARK. So in support of CFD, as we mentioned, Sahana is our program officer. So welcome, Sahana, and thank you for closing us out today.
Thank you, Jennifer, and thank you to all of you who made it through the full program. It's always, I was like, gosh, I'm the last thing on the agenda. Are people still going to be there? And it looks like you still are. So I'm so happy that you're here. so happy to see this event. It's been really wonderful to hear all of the projects that have come out from students and mentees this summer. And it's just exciting because this is what we were hoping to see last year, and now here it is. And so thank you so much for all that hard work and the presentations. I'm going to change rhythm a little bit, maybe change scope a lot, and talk about NIH more broadly. You've heard a lot about CFDE today, and it is one of many programs within the broader NIH ecosystem. And I thought it might be helpful to just kind of go over how we work, generally speaking, and how you can interact with NIH through applications for funding, for fellowships, et cetera. So that's sort of my goal for the next 15 or so minutes. And if there's time, I'd love to be able to answer some questions later on. So please move to the next slide. Thank you. Now, NIH is a really big agency, and I think you've been hearing a lot about NIH in the news. We've been noticing a lot of changes coming up. And so what I want to provide you today is just a little bit of context on what we're trying to do in general, and then provide a bunch of links out where you can go even later to look where updates are being made and kind of keep yourself fresh on what NIH is doing and the way you can connect with us. So generally speaking, on the left-hand side, NIH has money that we spend internally at NIH, and that's kind of the purplish section of that donut. That's about 20% of our budget that we spend internally on science, on personnel and things like that. And then the larger portion, which is about 80%, is spending outside of NIH. And that is where kind of CFDE fits in. So if we look at the outside of NIH funding in fiscal year 2024, it was over 35 billion. So it's quite a large amount of money. And all of that money is distributed across 27 different institute centers and offices. So these are distinctions made. For example, you've probably heard of NINDS or NCI, NIBIB. There are many, 27 of those different institutes and centers that have their own missions and that try to promote research, biomedical research related to those missions. And then if you look across NIH completely, the way that the funding is given is different types of awards. And the large majority of grants that are made are research grants. So that's 94%. There are also other types of awards. There are training grants, fellowships, construction grants. But basically, you're looking at a lot of the money going towards research project grants. Next slide, please. Now, we've already heard a lot about the Common Fund, and we've heard a lot about the Common Fund data ecosystem. So I'm not going to spend a lot of time here, but just to make the point that of those 27 ICOs, or institute centers and offices, the Office of the Director is one, within which is the office that administers the Common Fund. And that's where George and I and others that George pictured earlier are working. So this is one kind of one office that produces funding opportunities that you might apply for. Next slide, please. Now, generally speaking, there are, you can say, two buckets of NIH grant types. One is what we call parent announcements. So those are notices of funding opportunity, or NOFOs, where you bring your ideas. They're unsolicited. So we're not saying we want to hear about a certain thing. We're just saying bring us your best ideas. So they're investigator initiated. And there are parent announcements, which are basically standard. So not specific about the kind of science. It's more about the structure of the application that it's requiring. These can be research-based, so the R series. There's the K series, which is career development. There's F grants, which are for fellowships, and then training grants, which are research training. So these basically you apply to them based on what you're working on. They go through review, and you'll get a priority score and a percentile rank. And then from that point, kind of you move forward through the process of funding decisions once you apply. There's also another kind of type, which is special research initiatives, and that's where NIH staff write NOFOs or funding opportunities that are focused on special topics. So we're asking for you to send an application on a specific theme or a specific sub area of science. And this is kind of where CFDE fits in. Most of the CFDE funding announcements are of this nature. They're asking for specific types of science to be done. These can be PARs, PAS, RFA. These are all kind of acronyms to address different types of solicitations. There are cooperative agreements. Some of them are phased awards, like you get an award for maybe 2 years, and then you have to show progress of a certain nature. And if you kind of meet that milestone or the milestones, then you'll be given the second phase of money. If you don't meet the milestones, your award ends there. So there are kind of more unique ways of managing awards that can be done through these special research initiatives. And these often receive a priority score after they're reviewed, but they may not be percentiled. And then just in terms of a little vocabulary, if you haven't heard the term activity code, it is a code that we use to kind of tell apart different types of grants. So for example, an R01, K23, F32, the first letter is the kind of award, research, career development, training, etc. And the numbers are harder to understand where they come from, but you just sort of memorize them. So these activity codes will be something you see on the funding opportunity announcement. You'll see it on your notice of award. And it's kind of each of those activity codes often has a type of research that it supports. Next slide, please. Now, when you are looking for a funding opportunity, this is something that I want to emphasize is a point of continual development right now, but we are moving at NIH towards using the grants.gov website for searching for opportunities. So there are two engines, the classic and the simpler. What I have pasted in the bottom right corner is it's a screenshot of the simpler grants.gov website. So you can use that. There are some filters there that you can use to try to find what you're looking for. For example, opportunity status is one of the filters. And you might see that there's a forecast, it's open or it's closed. What that means is that a forecast means that something is coming soon. It hasn't been published yet, but keep your eyes open. There's going to be a notice of funding opportunity soon in this field. So start thinking about your ideas, start putting together your team, start imagining, what your application might look like. When the opportunity status is open, it means you're definitely able to apply. And there's going to be some receipt date, application submission date that you have to look for, but that one, is a live open opportunity. And then closed, it will be still listed, but it won't be, you can't apply to it anymore. It will give you some sense of what has been done in the past. So that's sort of, one of the filters there. You can filter on eligibility, on agency, and a number of different filters. So check that out. It might take a little bit of getting used to if you haven't used this in the past. But it's probably one of the methods that is going to be available to you moving forward. So please note that, especially if you've been used to using other methods in the past, try to take a look at simplergrants.gov or grants.gov from now on. Next slide, please. Okay, now if you're looking for funding opportunities within the Common Fund, you can also look at our website. So this QR code will take you to our Common Fund website where the active open funding opportunities are listed. So right now, or I guess as of last week when I put this slide together, and I went to that page, we only have these 4 opportunities open right now. But if you check back in say 3 months, there might be different ones, or 6 months, you might see something else. So you'd have to keep checking back here to see if there are any Common Fund opportunities you want to apply to. Next slide, please. Now, if you're looking for training opportunities, here are a bunch of links that you can reference as you're looking around. So as I had mentioned, at the very beginning, some of what NIH does is internal at NIH. So we have an intramural research program where there are, a huge number of intramural research labs where science is actually done on campus at NIH. There's also the clinical center where clinical research is done at NIH on campus. So that first link, intramural, takes you to the training and education opportunities there. I did my postdoc at NIH intramural, so that might be something of interest to some of you here today. And there are also faculty positions or scientist positions that are open there from time to time. So that's a place you can look for training and career development. There's the clinical research training and medical education. There's the Office of Clinical Research. There's trainings on there for clinical research if you're interested in that. And then there's the link for extramural, which is a link to all the different types of training grants that you can apply to that NIH supports. So this is the money that goes outside of NIH. So those 4 links are on that website at the very top that says training-opportunities at the end. But I'd also like to highlight, which is the purple rectangle, that the ORISE program, so Oak Ridge Institute for Science and Education, which Jennifer has been working with a lot. So actually, I would say if you have questions about ORISE, please direct them to Jennifer Burnett. But they have a wonderful NIH participant program. And the link is here, down here in that purple rectangle. When you go there, you'll see you can, there's a search engine there that can connect you with a whole lot of different training opportunities and career development opportunities at NIH. So that's an excellent resource for searching. A lot of these things as well, by word of mouth. So as you go to groups like this, meetings like this, conferences and meetings, and you start to meet people, you can also hear about training opportunities in other people's labs and such. So keep that in your mind to keep networking as you go.
Next slide. Hannah, I'll just mention real quick, since we did have a lot of faculty register, these ORISE programs are always actively looking for mentors as well. So this applies on both ends here. So if you have a need to, have interns kind of more consistently within your academic institution, this is a great opportunity to partner with NIH and many other agencies. So I just wanted to jump in and mention.
Thank you. I'm glad you did that. All right, next slide, please. All right, so when you are thinking about applying to NIH, there's something that I can't stress enough, which is don't forget to read the entire notice of funding opportunity. Unfortunately, and I apologize, they are very long. They are very long and they can be very dry. They can be overwhelming. You look at them and you think, oh my gosh, I have to put this down. I can't look at this today. And that's fine. Give yourself enough time. Put it down. Look at it tomorrow. Look at it next week. But do look at all of it because there is a lot of information in there. And there's generally like a template format that most of these NOFOs will take. So there's always going to be this table of contents, which I kind of screenshotted into here. Right now, it's just a screenshot, but in the NOFO, in the electronic NOFO, all of those section numbers are clickable. So you can just jump to whatever section you want to. And it really helps to navigate the NOFO when you're able to come back to the table of contents and start again. But there's a lot of information in them, which is why I'm stressing how important it is to read it. What is this about? Who can apply? What do I submit? What are the review criteria? And I think that is critically important because you're going to be reviewed. And on the basis of that review, the funding decisions are going to be made. So make sure that as you write, you're basically answering all the questions the reviewers are going to be looking for. And make sure you answer them in a way that they're going to find it easily. They don't have to dig too hard to notice, what it is you're trying to do. So many questions come up as you read this. Do they take clinical trials? Do they take resubmissions? Is my application responsive? Which IC is participating? When's it due? So take your time and read through this. And then when you think you've done it, go back through it. Do it with your colleagues. Make sure that everybody on your team who's applying is familiar with this. So that way, when you go to start putting together your application, you feel, more connected and you understand what it is that you're looking for. But as you're going, there's probably going to be some questions that you can't answer by reading the NOFO. So that's when you want to reach out to someone. So next slide, please. That's when you probably want to talk to your program official. So we call them POs for short. Sometimes people call them program officers. Sometimes people call them program directors or PDs for short. All of those are basically the same general role. It's a person like me, like George, who works at NIH and who is in charge of a certain portfolio or is able to manage program awards. So you can find us through using this really great tool on the NIH Reporter called Matchmaker. So this Matchmaker tool, and this slide, I guess I didn't put a link to it, or I can't see it at the bottom, but I know I have a slide in the future with a link. Oh, it is down there. Okay, I see it now. So what you can do in Matchmaker, and I really, really love this tool, you can basically copy and paste a summary that you wrote of what you want to do, or what your specific aims are. This is what I plan to do in this order. So copy and paste it into that text box. And on the right-hand side, you see that little button that says similar program officials. Make sure that that's checked and hit search. And what it's going to do is it's going to bring you back a list of projects that match yours pretty well in terms of topics. And it will tell you which institute funded it, who is the program official, and so much more. And it's abstract, that's public on Reporter. So you can get a sense of who is managing the awards that are similar in nature to my application. Based on that, you can reach out to those program officials. And the kinds of things that you might find yourself talking to a PO about are grant policies. Is this allowed? Is that not allowed? When do I have to do this? What's the procedure? How do I change my institution? Look at these cool new things I'm doing. I'm so excited. I want to share them with you. Program officers, often we were previously in academia. Maybe we were faculty, maybe we did a postdoc, maybe we worked at a company doing research. So we would love to talk to you about your scientific advances. So this helps us to learn the field, and then hopefully we can help you to navigate getting funding within NIH. We can provide feedback on finding NOFOs, or is this NOFO appropriate for what you want to do? And the list goes on and on. So this is just sort of a snapshot discussing your summary statement after review. You've got an award now. What happens next? If you need supplemental funding. So these are the kinds of ways that you might want to interact with the program official, either before award, during application time, or after award. We can try to be as helpful as possible in that range. Next slide, please. Sometimes it's intimidating also to reach out to a program official, especially if you don't know them. So I'd like to state and assure you that though sometimes POs might be really busy, our job is to help you. So please understand that you have every reason and right to reach out to us, and we will get back to you. So this is my recommendation to you if you're reaching out for the first time and you're not sure how to do it. I say reach out by email because in that email, you can provide a little bit of information up front, which will give the program official a little context to figure out, okay, I think I want to set aside 45 minutes to talk to this person, or I already know from what they're saying that they should really talk to him or her in this other institute because I'm no longer working in that area. So we can help you like as best as we can in a shorter time frame. So email us, send some information like your bio sketch if you have it prepared, and your research goals, like a draft specific aims or a summary that you're ready to share out. Ask a question if you have it. If you feel like there's context to say, like certain deadlines are happening professionally that you need to, this has to happen in a certain time, just be clear about them so it's easier to plan. If you're looking at certain NOFOs, mention what they are, because the PO might say, well, you didn't look at this one, or actually that one's closing, it's not going to work. So, provide as much information as you can, and then request a time to talk. So when you actually get on the phone together, or Teams, or whatever it is, be prepared to give like a 5 or 10 minute summary of what you're doing. Sometimes people provide slides to talk through it because it helps them, especially if there are a lot of diagrams or images or something that you can't really talk about, but you don't have to. You could just talk to them. And usually that'll just take on its own life, and you'll see where that goes. And hopefully it would be helpful for you. Next slide, please. Now, once you put in an application, what happens is it gets reviewed. So I just want to make a point that the peer review of NIH applications is separated from the funding decisions for NIH awards. So program staff like me, we are in that like right-hand purplish bubble, NIH Funding Institute Center or office. But when you first apply, your application goes to that left-hand side green bubble, which is the NIH Center for Scientific Review. So you apply, the Center for Scientific Review will receive your application. They refer it to where it's supposed to get reviewed. And then scientific review officers at CSR will lead the review, the peer review of your application. So during that period of time, really the scientific review officer is the person in charge of your application. Once they finish the review and they produce a summary statement about 30 days after the review, then the program officer becomes the person who is in charge of your application up to potential award. So something just to remember, who's going to be holding your application depends on what point of the application cycle you're in. Going back to CSR for a minute, a lot of people ask, well, what study section is going to review my application? And I love this tool that CSR produced. It's called the Assisted Referral Tool. It's the link in the center of that green blob. If you click on that, you can input, you can paste in your specific aims or a summary again into it, and it will give you back kind of a ranked list of different study sections that are relevant to your project. So you can look at the websites for those study sections, and it'll give you a sense of the composition of the reviewers. Are they physicians? Are they engineers? Or, or are they radiology background, or whatever it is? You can take a look at the focus of that study section and just take a look at the fitness of your application to that section. And then another thing to point out is that the review, the CSR, has been working to simplify the review framework. So if you've been applying for a number of years to NIH and you submit a new grant application soon, you might notice that the review criteria have shifted. It's worth looking at in advance before you submit your next application. So for a number of different activity codes, it kind of got reduced from 5 areas of review to 3 factors. So previously there was significance, innovation separately. Now they're considered together in a factor called importance of research. The rigor and feasibility factor now is basically what we used to call approach. And then the factor 3 of expertise and resources combines expertise and resources, which were reviewed separately before. So be sure you understand how your application will be reviewed so that you can produce an application that matches that. And let's see, what else did it? And then the website for CSR is there under the NIH Center for Scientific Review. You can take a look at that, spend a little time to understand how they're organized, just for your information. Next slide, please. Okay, so I mentioned Matchmaker before, and I'm bringing it back here because I wanted to make sure that you knew about the tool in a broader sense. So it's called the NIH Reporter, and it is a public tool where you can take a look at what NIH has been publishing and is actively publishing, funding right now. So here's a screenshot of some of the different features on the NIH Reporter. You can look at active funding by state, by institute or center. You can do an advanced project search with all these different keywords and ID numbers and names to figure out, if you're trying to find something very specific, like in the year, 2013, what was funded in this topic. You can do those kinds of searches here. I showed you Matchmaker, and there's also a publication search. Something I want to mention about Matchmaker, I mentioned it before in the context of finding a program official, but you can also just spend a little bit of time playing around with it to look at what else is funded, what universities got funded to do similar research, who are they, what was their goal of their study. So it'll give you a lot of context on the community that's working on similar research as you. You might recognize some people, you might not, and you might wonder, okay, why don't I know about them? And then you can start looking at the publications. So in the publication search, every grant award has to report on publications and outputs of the program. So this is a way to understand what publications have come out of NIH-funded studies. That can be really helpful. You can, poke around there and try to see, where does your idea fall in the realm of what NIH has already been funding? And that might help you figure out your next steps as well. Next slide, please. Now, finally, I want to share this website with you, grants.nih.gov. And one of the reasons why I want to spend a minute on this is because there are a lot of policies that are kind of in flux right now. This is a really dynamic environment. Whatever I'm saying today is correct today. And I expect it and I hope that it's correct in the future. However, there might be updates and there might be additional steps or, slight modifications. So this website has a number of, on that blue banner, it has a number of different sections. I really do recommend spending a little time with this website because, especially the new to NIH, it goes through a lot of what I said, but in great detail. It'll take you through the full process of how to apply. It provides a ton of information and resources. Then there's the funding and grants process. You'll find a lot of good resources there. What I'm calling out here in policy and compliance, there's a section where we have new initiatives and policies that are like constantly being implemented. So if you're interested in what's the next new thing, there's like a section where there's a look ahead, like we expect this to happen soon. And there's like, this is what's happening right now. And you can look back into history to understand what is the current policy. In the news and events, I think you'll find a lot of helpful links to get you kind of connected. So there is the Extramural Nexus, which is kind of our newsletter. You can look there for updates on the system that we use for grants. There are on-demand event resources. You can subscribe and follow in that section. There are podcasts that you can use to keep yourself updated on especially new policies that are coming your way. And you can also always reach out to me. So, I always offer myself as a resource when I talk to groups like this. So please do let me know if you have questions, and I'd be happy to talk to you or give you a hand navigating our websites and figuring out how you get where you need to go. I hope I didn't take too long, Jennifer. Thank you so much for your attention, and I'd be happy to answer questions if we had time.
Thanks so much, Sahana. No, that wraps us up. I will say, luckily, I think it only affected the ORU staff. Our network glitched there at the very end. So I apologize. We may have missed just a very, very minute moment of recording. But Bo, if you're able, we can move to the closeout slides really. Okay, so first and foremost, your feedback is extremely important to us. So please take a moment. It looks like I'm unstable. I'm going to turn my video off, hoping that everyone can still hear me. Please take a moment and fill out our evaluation survey. We really want to hear from you. And if someone from my team could put the link in the chat, if again, folks are online, Yeah, oddly enough, I think it's only our ORU network that is being affected here right at the end. Yeah, so a few items to close out here. You can learn more about all of the work that the CFD Training Center is offering by visiting our website. Look for our mentor program again, that application coming out this fall, which is actually only a few months away. So keep an eye on our website coming up in October, November timeframe. Please take a moment and join our e-learning dashboard or share this information with your students. The how-to series in particular provides a great intro to broaden research interests and provide information on relevant CFD data sets, especially those looking to just get started. Check out our upcoming events. We're updating that page frequently. And then finally, if you're interested in collaborating with the CFD Training Center to become a mentor, want to share about your research on our podcast, or want to join our Decoding the Data Ecosystem Collaborative, which we are looking to expand beyond the mentor program and more into a collaborative, research and discussion, please reach out to us. And with that, really appreciate everyone sticking around for the symposium today. We will have the recording available sometime next week. And we also will share as much as we can of the slides, and we'll have all this information up on our website as quickly as we can. So thank you for the extra 3 minutes. And I'm glad that our tech issue was just there at the very end. So thank you again, everyone. We really appreciate your time. Thank you.